test it.
Oh yeah, okay.
Bonus. C'est le Tintin.
Ouais. Dans les Tintins, il y a des personnages de plus.
Testing.
Méchant.
It's okay, John. Hello.
1, 2.
I think somewhere around here is best, and then that's in a pocket. Then
But how to...
yeah, you just put Hello.
I take one micro.
Huh?
I take one micro.
One. So now that, yeah.
We should try to help people.
Yeah, I'll push people through now.
Can be really short. So.
Yeah. So, because these ones to go through the slides, and now going back is okay now if you want to do it, and then this one in here for the pointer.
Yes.
Okay.
Yeah.
Great, thank you.
Yeah, is that okay? Yeah.
Yes.
So you probably need to have an introduction or something?
Oui,
for
je
the-
dis juste ton nom et le titre de ta présentation et après tu...
C'est juste pour moi, pour la séance, pour le,
Oui,
pour le reste
je
aussi.
vais dire pour la séance, je vais dire aussi un truc par rapport au streaming. Tu as déjà tourné tout ça. Et après, donc
Ok.
je dis la session, on ouvre la session devant toi et après je dis ça. C'est bon? I have my sheet notes. C'est le programme parce que j'ai pas envie de faire des conneries.
T'es jeune, hein, tu peux le voir.
Ouais, jeune, hein. J'ai des lunettes depuis que j'ai peut-être 10 ans, hein, donc tu vois, ça va rien dire.
Good? I can take this one from the back.
Hi everyone. I hope you slept very well because today we are going to have a great session which is more about the work package which has focused on human response. So the title of the session is Functional Genomic Basis for Human Responses and Disease Resistance in Farmed Fish. So just before to start with Costas, just a note again for the people which are following us in streaming that they can still ask a question on the chat and we will try to make the speaker be aware about it and reply to it. Okay, so today we are going to start. Our first speaker is Costas. And his title is Genome Functional Annotation of Host defense response in sea bream. Yeah. So the floor is yours.
Thank you, Mo. Good morning, everybody. I'm Kostas Tsigianopoulos, coming from the HCMR in Crete. So first of all, I have to clarify that I'm not an immunologist. So I did my best to learn the lesson that my scientific partners prepared. So just a few words to see that the sea bream together with the sea bass are considered the two most important species for the Mediterranean aquaculture, constituting something like 80% of the production. This is an industry that generates each year more than $1.6 billion of profit, and gives work to thousands of people in the Mediterranean area, and not only. For the species, ever since the species was introduced into the aquaculture industry, there are lots of studies, first in population genetics, and more recently concerning the genome assembly, which was first done by Pauletto et al. In 2018, so a joint effort of HCMR with the University of Padova. And a year later, it was the first genome also that was deposited in the gene bank by the VGP project. Even though we have able transcriptomic data, we have few recent epigenetic or epigenomic studies that have to do mostly with the feed replacement. And for the epigenomic data, we have practically no information. As I told you, I'm not an immunologist, so the purpose was to see in the work package 3 how the fish are reacting to a stimulus that is delivered either in vivo or in vitro. Just to remind you that we have from one part the innate immunity system, which is composed of the skin, gills, gut, and all the mucosal structures that are supposed to keep the parasites, the viruses outside of the organism. And as soon as the pathogens are getting into the organism, there is the adaptive immunity system that starts to function through the beta and T lymphocytes. So into this project, we were mostly focused studying the head kidney, which is considered to be one or the primary organ for the immunity response to the pathogens. And concerning pathogens, unfortunately, the species has a lot of pathogens that is affected by. So we may have a bunch of bacterial diseases that are affecting the species. We have viruses, and the more recent one being the reassortant beta nodovirus. So a kind of new reassortant strain between the red grouper and the striped, the striped jack nervous necrosis virus, and parasites like microsporids, enterospora, enteromyxum, and other bigger parasites like the monogeneans, Sparicotyle, Chrysophori, which is a parasite that attaches to the gills and is provoking anemia to the species. So the aim was first to conduct the first study on gene expression coupled with the immune response in gillhead sea bream using RNA and ATAC sequencing data. Mo and other colleagues yesterday explained more or less what was the the design of the experiment that was based on standard operating protocols and procedures that were optimized into the AQUA-FAANG project by the Dutch, the Scottish, and the Norwegian partners. So from one hand, it's not exactly the best representation, but from one hand we had fish which were injected with the pathogen, which was either an attenuated Vibrio strain or a PIC, chemical which imitates, let's say, the viral infection. And afterwards, we were taking the head kidney, the leukocytes of the head kidney in order to do all the downstream library preps. And on the other hand, we had the head kidney first to get the leukocytes and prepare Petri dishes on which we put the pathogenic PIC or the Vibrio. For the bioinformatic analysis, I again apologize. It's too complex for me, mostly because I'm not a bioinformatician. So we employed for the RNA analysis the NF core. And of course, we used the reference genome to map the sequences in order to quantify the differential expression of the transcripts. And more or less, it was the same that we used with NF core in order to study the differential accessibility with the data coming from the ATAC libraries. What was really interesting was the first essay to combine the two types of data, which is, of course, going to be completed with the addition of the ChIP-seq data later on, because it was data that were generated earlier in summer 2023, so we need more time to do that. So in that case, we identified peaks of the ATAC-seq, and we tried to study 1 megabase region down and upstream of this peak. For the RNA sequencing, for each experimental condition, we generated more than 400 million reads, and approximately 65 megabase pairs of data. And we can see in this slide that we have a very big number of differential expressed transcripts concerning Vibrio. But when it comes to the in vivo experiment in which we injected this pathogen into the organism, the number drops very dramatically. Between the experiments, okay, what has meaning is, of course, for each treatment, the in vivo and in vitro comparisons. So when we study the Vibrio, we see that concerning the upregulated genes, we have 39 and only 41 downregulated. And for the PIC, we also have some hundreds of transcripts upregulated and very few downregulated. In these volcano plots, we see the upregulated transcripts in red and the downregulated in blue, and the same for the in vivo experiments in orange and green, respectively. If we could also schematize that with the circles and the Venn diagrams. It's more or less what we showed in the previous table with the transcript in common with the overexpressed and down-expressed transcripts. Here we try to represent the assembly, the genomes, and the chromosome of the gill-head sea-bream genome, and we see that the Vibrio in vitro has lots of transcripts differentially expressed compared to the in vivo, which has very few. This is the let's say the almost white inner circle, and lots of transcripts concerning the PIC distributed around the genome of the species. Now, if we take a look at the genontology terms of the transcripts that have shown a differential expression, we spot genes that are really involved in the immune response and the metabolic processes, and have to do with stimuli from external conditions, biotic conditions, or defense response, and so on. This is also the case when we take all the treatments differently, and we have overrepresentation of immune defense terms, as well as metabolic and catabolic processes. If we go now to the ATAC results, it was quite surprising that for the in vitro experiments, we have very, very few differentially expressed, differentially accessible peaks that showed to be significantly supported. We have, for the in vitro experiment, only 14 peaks out of the almost 200,000 peaks called. For the in vivo experiment, it showed almost 3.5 thousand differentially accessible peaks out of the 252,000 peaks. And this was quite surprising. I didn't have the time to prepare the same slide for the RNA-seq, but it's quite interesting to see that concerning the in vitro experiment, we see a clear clustering of the control, the PSC, and the Vibrio, let's say, cells that we prepared with the pathogen. Whereas when it comes to the in vivo experiment, we see a more scattered distribution of all the 54 samples, 18 by experiment. As we saw for the in vitro experiment, we have practically no volcano plots that can show the real situation. And here, as I told you, we have very few differentially accessible regions for the in vivo, most of them concerning the PIC factor that was used. Now, if we are going to make the coupling of both approaches, we see that we have we have a potential enhancer activity of peaks with neighboring genes. We identified silencer activity with upregulated genes next to under-accessible peaks, or down-regulated genes next to over-accessible peaks. And we saw that in most cases we have to do with a response to interferon gamma. And since we don't have time to talk about all the differences that we found for the RNA-seq experiment, we conclude by saying that We have upregulated genes next to under-accessible peaks, which really showed an overrepresentation of the IFN gamma response. And we know that interferon gamma is a primary activator of macrophages, so it's more or less normal what we saw, stimulating natural killer cells. And these are the main conclusions. So we are happy to be part of this joint effort. We have worked a lot, but we have also been lucky and working for the first time with these approaches, the ATAC-seq and ChIP-seq. I also like to thank the coordinator of the project, Dan, Matthew, and especially Luca and his team, because we collaborated a lot and exchanged the protocols in order to optimize. And it's still the ChIP-seq integration that remains to be done. And these are the main collaborators. So it's let's say, the bioinformatic people with Teresa Manoussaki, Machi Papadopoulou, and Vasilis, and all the people that have really worked hard in the lab to provide all these results. Thank you.
So thank you, Kostas, for this amazing talk. Is there any question for Kostas? I have some. I have one at least. Do you think that the fact that you don't see any differences in the ATAC-seq is coming from problem maybe on the sampling, so that it's not actually each kidney that we should figure out what are the signals, but in other, as you said at the end, other human cells like macrophage that should be purified and checked?
I'm also very curious to see what has happened also to other species. That's why I'm waiting the CBAS results to see also if in the other species have similar results, and if it's a method to follow in the near future, let's say, or we just may skip the ATAC and go to the ChIP-seq if it really gives you much more information. So instead of investing months and of work and consumables, go for the ChIP-seq. But I don't really know the biological answer behind that. But I would like to see the TERBUT and the CBAS results.
It's coming. Okay, thank you, Kostas.
Thank you.
So our next speaker is Serena, and she will present a talk about the transcriptome and chromatin landscape of CBAS, immune response to viral stimulation.
Thank you. Can you hear me?
Sounds good. This is for the laser, and then to go between slides.
Okay. Thank you. Good morning, everybody. I'm Serena from the University of Padova. And today I'm going to talk to you about the transcriptome and chromatin landscape of European CBAS in response to viral-like stimulation. I guess the European CBAS doesn't need an introduction here. It's one of the main species of a major economic value in Europe. Its farmed production has greatly increased in the last 20 years, from 7,000 tons to over 200 tons. But the disease outbreaks that to date involve around 10% of all fish mortalities in farms, represent a severe threat that can interfere with the progress and the sustainability of the aquaculture system. That's why an adequate knowledge of the molecular mechanism that control the host response to pathogen is of crucial importance to understand the genome function and regulation. And in this context, the epigenetic landscape in terms of chromatin accessibility to the transcriptome machinery and the histone modification play a key role in transcriptional regulation. As part of the AQUA-FAANG project, and in particular within the Work Package 3, we aim to profile the in vivo and in vitro response to European CBAS in response to stimulation with a viral-like mimics. And we aim to provide the first genome-wide functional annotation map of the European CBAS. As Kostas already explained, our experimental design involved two different experiments. One in vivo stimulation, in which adult naive CBAS were divided into two groups and stimulated by intraperitoneal injection with either PBS or poly-AC. The animal were then sampled after 24 hours, and the head kidney was taken for further analysis. For the in vitro stimulation, even in this case, the head kidney from adult naive CBAS were sampled and employed for the isolation of the myeloid cells. Then the isolated leukocytes were divided into two aliquots and stimulated either with PBS or poly-AC. After 12 hours of incubation, the cell cultures were sampled and employed for the further analysis. What did we do? First of all, we carried out RNA-seq analysis in order to profile the gene expression in poly-AC stimulated samples compared to controls. Then the same samples were employed for ATAC-seq analysis in order to define the region of chromatin accessibility across the whole genome. And then the ChIP-seq technology was applied targeting three different histone modifications: H3K4 trimethylation that correlates with promoters of active genes, H3K27 acetylation that marks active regulatory elements such as enhancers, and H3K27 trimethylation that marks actually genes that have been repressed. Then we applied an integrated bioinformatic analysis in order to define how the chromatin states can control the gene expression. Focusing on gene expression, first in the in vivo experiment, the principal component analysis highlights a clear separation between the poly-AC stimulated and control groups, and a pairwise comparison analysis carried out with stringent criteria allowed identification of 1,500 differentially expressed genes, almost equally distributed between upregulated and downregulated genes in poly-AC stimulated animals compared to controls. The overrepresentation analysis was conducted in order to find the gene set or biological pathway enriched in differentially expressed genes, and we did also a gene set enrichment analysis in order to find or to define biological processes or gene set that were enriched in genes showing the same changes in expression between groups. Both analyses gave concordant results, with the most significant enriched gene set being actually custom reactomes. Those reactomes are a list of genes found in recent literature and involved mainly in immune response, and in particular response to viral infection. I don't know if you can see from there, but those ones are the ones reported by Pereiro and are genes differentially expressed in European CBRIM, in gilted CBRIM, sorry, in response to necrosis nodavirus infection, while the one reported by Levrault are genes upregulating following injection with interferon or chikungunya virus, while effector ESG are actually interferon responsive genes that are the ones that drives the antiviral state. Only two gene sets were found enriched in downregulated genes. They are partially overlapping, and they are composed by inflammation-related cytokines. As for the in vivo, also for the in vitro analysis, the principal component analysis highlighted clear separation between poly-IC-stimulated cells and controls. Even if I have to agree with Kostas in this case, if you look at the hierarchical clustering, the cells tend to cluster according to the fish they originated from. And we appreciated that even in the ATAC-seq analysis. Anyway, the pairwise comparison allowed the identification of 648 differentially expressed genes, 380 upregulated, and 268 downregulated in poly-IC-stimulated cells compared to controls. Even in this case, the overrepresentation analysis highlighted a great increase in significant gene set among the ones involved in immune response. Most of them are shared between the one significant in the in vivo experiments. So the next step was the integration of the ATAC-seq and ChIP-seq data in order to define the chromatin states in the epigenetic signature of poly-IC and control samples. We applied the chroma-HMM software in order to define each chromatin state based on the presence or the absence of the ATAC-seq and histone-modification signal and their combination. We were able to define 10 states that we labeled with a functional meaning, such as active promoter, enhancer, repressed polycomb region, or quiescent region. What we observed was a difference in the distribution of those regions in response to poly-IC compared to controls, but also in vivo samples compared to in vitro. But we wanted also to see if the chromatin state correlates with the gene expression. So we had a look to the coverage, so to the depth of coverage of the RNA-seq signal in those regions. What we could see, and it was a confirmation of the right classification of the chromatin states, the active regions show a significant higher expression levels compared to the regions that were classified as repressed or quiescent. You can appreciate here very well. Here, I think you cannot read, but those are active promoter and enhancer compared to quiescent region or repressed regions. Then we focused actually in the changes of chromatin states in poly-IC stimulated samples compared to controls. We first of all looked at the repressed regions that showed actually an increase in their coverage in poly-IC stimulated samples compared to controls. The regions located, the genes located in those regions were actually not expressed, so that we expected that. We didn't find a significant increase in the proportion of downregulated genes in poly-IC compared to PBS. That's why the great majority of them, even before stimulation, were in quiescent regions. And we didn't find any gene set significantly enriched. When we looked at the genes that got activated after the stimulation, the situation was dramatically different. We found a significantly higher proportion of upregulated genes. And the overrepresentation analysis found as significantly enriched gene set mainly involved in immune response and interferon responses pathway. And of particular interest are the main induced genes, so the genes that got activated after poly-IC stimulation and that show the greater fold change difference in poly-IC stimulated genes compared to controls. Between them, many are interferon responsive genes, and I would like to focus your attention on those one, interferon alpha inducible protein 27 and so on, because Robert will talk about it soon after me. They are actually located in a major QTL that is significant for the CBAS resistance to necrosis nodavirus. The same goes for the in vitro experiment. When we looked at the genes that got activated after poly-IC stimulation, we appreciate a significantly higher proportion of high-regulated genes. Even in this case, we found the enrichment of pathway involved in immune response and in interferon responses pathway. And so having a confirmation of that the chromatin states correlates then with the transcriptional regulation. So to conclude, the present study provides a first genome functional annotation map of the European CBAS in response to an immune stimulation. The differentially expressed genes between conditions highlighted an enrichment in immune biological processes, in particular to interferon related pathway. And we identified significant changes in chromatin states that correlate with the transcriptional response. What we are going to do now is just to analyze the transcription binding factor binding sites that are enriched in those differentially activated promoters, and we aim to match the regulatory elements we found in our experiment with genetic variation. I would like to thank my colleagues from the University of Padua that did a great job, and all of you of the AQUA-FAANG project for a always helpful discussion and suggestion. Thank you.
Really nice. So we have plenty of time for questions. I'm a really lucky chair today. For Serena, so please go ahead. Yeah,
thanks for your talk. I really like the approach of the hierarchical clustering, where you're just showing that whether you know the function already, and then you're validating that with Go categories. But I'm wondering whether, can you link the ones that previously didn't have an annotation to an immune function? Can we link them to now know that you've defined them as Go terms that are linked to immune function, where they didn't previously have one?
I'm sorry, I didn't listen.
Where you have your hierarchical clustering shows a group of genes that may not be annotated already to have an immune function.
Yes.
So can we now make that link and connect, like give those genes that we previously didn't know what they do, can they now be attributed a function in immunity through a Go category term or something like this?
Where did you, I'm not sure I understood your question. So I'm really sorry, but I don't, in the heat map, okay? In this one?
Yeah, so the heat map, you'll have some genes there that don't have a Go category annotation, but they're clearly involved in the immune system function.
Yes.
So I'm wondering, and it's probably a broader question than just for your study, but can we now link that gene, which previously had no annotation, with no Go category, can we now make a link to say that it has a function?
Yes, but those are all annotated genes, actually. Not all of them have an immune function. There is a great increase in enrichment in immune-related genes, but I'm sorry, I think I didn't understand your point.
I'll talk about it later. I think
yes,
it's a question for everybody, is how do we link the unknown function of genes with, you're validating the fact that these Go categories are enriched, but there are genes in there that don't currently have an annotation.
Yeah.
And can we now make that link, because you have all this foundation of fundamental evidence to show that
course.
they're involved in immunity, but we previously didn't know that, if that makes sense.
So actually I can say that this is a great point for the discussion time that we will have this afternoon, where we try to figure out what can be the next step of those projects, and actually indeed we are looking, all of us, for better annotation of genes, especially in the different species, which has been involved in AQUA-FAANG. So, yeah.
Hi. Sorry.
Paulino, yeah.
Hi, Serena. Very nice talk. Thanks. I have a short question. You presented the PCAs separately.
Yes.
Have you the picture of the all conditions in a single PCA? They are the four clearly separated or not?
In vivo and in vitro are clearly separated. When you put everything together, the two different sources are so polarized that you don't appreciate very well the difference between groups in the same experiment, actually.
Okay, thank you.
Hi, really nice talk. I just had a really quick question, which was you presented the poly-IC data. Could you give us like a quick oversight of how the Vibrio data looked in CBAS and what your plans are?
Yes, unfortunately for the in vitro experiment, the Vibrio data were not good, in particular for ATAC-seq libraries. So we had a big problem with the Vibrio stimulation in vitro experiment. For the in vivo experiment, it was good, and even in that case, we had a great response in differentiate in the gene expression analysis with immune-related pathway enriched. We are analyzing them now.
Okay, thank you. Thank you a lot, Serena. So as Serena mentioned, so now we are going to have Robert, which will continue to talk about CBAS. And his title is Functional Genomic Architecture of Viral Nervous Necrosis, Disease Resistance. And of course, he will present his famous QTL. So really interesting for the industry here.
Just get it to grip on there. That's it. There you go. That should be fine. Put that in your pocket. The pointer's just the red one in the middle there.
Okay. Thank you, everyone, and I'm happy to speak after Serena because, yeah, most of what we have here is very related to the experiments that they did in the study that she has presented. So most of the introduction is not very different from what Serena has said. So CBAS is really an important aquaculture species in Europe with that value that is up to almost $500 million USDs in terms of exports, and that Serena has indicated. And through the recent years, there has been a quite gradual increase in production, and yeah, like today, it's mainly aquaculture production that is accounting for most of the CBAS that is consumed in Europe. So, as you know, that intensive aquaculture production exposes the species or the fish to different environmental stresses, and that includes diseases. And for European CBAS, vibriosis and VNN are quite the main disease challenges to the aquaculture industry. So VNN is caused by a positive single-stranded RNA virus, and that virus mainly targets the nervous system or the central nervous system, which includes the brain, the spinal cord, and the retina. And some of the target species include the European CBAS as a species of interest we are talking about today, but also Costas highlighted that it is also a major problem in sea bream. So this is really a major problem, especially for the juvenile fish, that can lead up to 100% of mortality of the fish. And recently, we are seeing that indeed resistance to this disease in farmed European sea bass is heritable, and there are studies that have including studies from our own group, have identified a major QTL on linkage group 12 or chromosome 13 that indeed accounts for a significant amount of genetic variance of the disease and of the resistance to the disease. And a number of candidate genes have been identified, but as you can see, different studies are identifying quite different genes, indicating that until now we are not very sure of the precise molecular genetic background that is conferring or that is modulating the resistance to this disease in farmed European sea bass. So that's where I think the dream for this study came from. And we employed quite multiple omics tools to see that we try to dissect this complex genetic background of VNN resistance in farmed European sea bass. So briefly about the materials and methods. So we had this huge population of sea bass, juvenile fish, which were generated from full factorial crossing. And we generated that population which was challenged with VNN for around 26 days. And another portion of that group was used in transcriptome challenge, still with VNN and then with a buffer. And then we generated RNA-seq, ATAC-seq, and ChIP-seq, which were analyzed for functional characterization of the genetic background of VNN resistance. And then the phenotype of the disease was recorded as binary survival, died or remained alive at the end of the experiment, or days to death, meaning the time that took the fish to die. So going to the results, we see that the crossing to generate offspring was quite well, and the study population indeed came from all the dams that we had and also from all the sires. And then we see phenotypic differences within the sire families and also the dam families in terms of variability in VNN resistance. As you can see that some of the families, the death was very rapid, while the others remained, only a few individuals died. So looking at the additive architecture of the trait, we see that indeed there is a lot of additive genetic variance of the trait, which is good news to the breeders, that we can indeed employ selective breeding to improve or to increase resistance and limit disease outbreak on the farms. So going to the GWAS. So going to the GWAS, we had quite a strong signal and quite close to the previous studies. And that major QTL that is located on LG12, almost all the significant markers came from there. And we had a few quite not very strong, but still significant signals on LG8 and LG1A. And we we indeed saw that this QTL has major effect, that one of the most significant SNP markers that we identified accounts for almost 40% of the additive genetic variance of the trait. And you could clearly see that there is a difference if you look at this bar plot. You see that indeed there is an additive effect of the QTL on the trait. You could see that individuals with a resistant QTL had a significant difference with those that had a susceptible genotype for the QTL. So you could see that indeed the individuals with a resistant genotype, as I have highlighted, had a higher survival rate when infected with the virus. Now, we went to look into the region where the most significant variants are located. And the region is quite complex, but also interesting, as Serena has highlighted. So this region of about 10 kb, it's kind of variable between the two annotations, so NCBI and Ensemble annotations. So we see that in the same region, NCBI indicates that there is a cluster of interferon-inducible genes, but in the same region, NCBI indicates that there is this very long galactase 8 gene. However, when you look at the RNA-seq kind of evidence, this gene model is not well supported as it is for the interferon-inducible genes. So now we had to work out on this problem, how do we go beyond this, and how we work out which of the annotations is really reliable. So we have this region from the previous slide that's shown. So we got a data, and that was Serena's work. So we got ChIP-seq data and attack-seq data from the poly-IC challenge, and then we worked out the gene models that are supported by these two sources of functional information. And indeed, we saw that the ChIP-seq and ChIP-seq RNA-seq data sets indeed supported the existence of these gene models from NCBI, and also the existence of this gene model from Ensemble. So what we did, we merged the two annotations, but we removed this gene, which is not supported by the functional information. So we now had a merged annotation that we worked with for functional characterization of the QTL. So we performed eQTL analysis, and indeed we got quite an interesting signal, which is exactly overlapping with the QTL. So even if the signal, it varies a bit with the genes and also with the condition and also the tissue, but this signal is overlapping together. So the most significant SNPs we are identifying here, or variants we are identifying here as the most correlated variants with gene expression, are the same variants that are associated with VNN resistance, as shown in the GWAS results. So we now wondered about what is the probable causal effect of these EQTL, QTL SNPs especially on the genes where they are located. And still Serena and Max from Padova employed the ataxic analysis, and then we identified 4 regions, 4 open chromatin regions within the head kidney, and they are overlapping with 3 open chromatin regions within the brain that are also within the QTL region, and they are overlapping these 3 of the interferon-inducible genes. So what is interesting is that 2 of the SNP markers are having quite a regulatory effect, which means they affect transcription factor binding sites. And that is the putative kind of regulatory effect of the QTL that we have so far, though this is coming from bioinformatic analysis, which is yet to be confirmed through functional experiments. So then we went into the literature to look at the function of interferon-inducible genes as they relate to viral immune response. Indeed, the function is not well defined. However, we found quite an interesting paper that they looked at quite a similar virus, which is a West Nile virus. It is an important human health virus, but it is also affecting the same tissue. It targets the central nervous system, and they clearly indicated through a gene knockout that this gene has got a significant impact on resistance to a West Nile virus. You could see clear difference between the knockout and wild type in terms of resistance here and also in viral load. So functions to think about, considering all the interferon stimulated or induced genes, as Thomas talked about yesterday. So they are quite involved in the number of antiviral processes, which we will have to study maybe in AQUA-FAANG too, hopefully. And we will have to precisely explore the mechanism through which this gene is modulating VNN resistance. So just to conclude, so we have sufficient evidence that indeed VNN resistance is a trait we can breed for. There is enough additive genetic variance to support that. And we have a major QTL, which can support even without going for the genomic prediction. You could go for marker-assisted selection. You have 40% of additive genetic variance explained, and you have 90% survival, over 90% survival rate of resistant individuals. So, yeah, our most probable candidate for modulating VNN resistance is indeed the interferon-inducible gene, and our data supports a kind of a putative causal mechanism of modulating the transcription binding factor site. I would like to thank all the members that have participated in the project, and also thank Joe from Dan's group. He contributed a lot calling the SVs, or the structure variants, which have also been added to the study. And yeah, they are quite interesting because also the peaks are there of structure variants. Thank you, everyone.
Thank you, Robert. Really nice to see that there is this amazing QTL. Maybe we have some industry partner which can be interested in this question to Robert or others. Yeah, Wendy?
Yeah, very, very interesting and exciting, of course. So you've talked about different tissues, but what is the actual cell type do you think this regulatory variant is playing out in?
Yeah, I think that is one of the things that we are trying to pursue. And because we have bulk RNA,
Yeah.
we are not very sure, but I think that is something we have to look into.
If you did some RNA fish or spatial
Exactly.
transcriptomics in tissues
Yes,
to find out,
yeah,
that'd be key.
yeah. Probably a single cell, good one.
So-
Somebody else? Who?
Thank you, Robert. Very interesting and clear talk, so that's really good. So I was kind of very impressed by your mapping resolution. So I think you said you had this QTL mapped within a 10 kb region.
Yes.
But when I saw your initial plot, it was almost the whole chromosome. So how did you define this narrow region to a very, very narrow region? It's 10 Ks, really small.
Yes, okay. We did quite a preliminary analysis, which I've not indicated. So we had a correlation between gene expression and resistance to VNN.
So it's not just the GWAS result narrowing it?
No. So it was a combination of things. And indeed, the genes that are correlated with the VNN resistance are located exactly in that. You could see all the other genes down.
Yeah, a last question from that.
Nice talk, Robert. Thanks very much. Given the difficulty of the annotation in the region, I was wondering if it might be a good idea, relatively easy, to do some long-read RNA-seq, like nanopore full-length RNA-seq, and you could possibly do that between the resistant and susceptible individuals. That might give you a better annotation of a very confident annotation of the genes, as well as possibly some insights. That was just a comment, really. But my question was, is industry already using marker-assisted selection for this? I was wondering how far along that actually was for CBAS.
No, before, no, but now we are working with Select if someone is here. We have already because they are partners in the project, so we have already provided the SNP markers to them to add them on this. They have a small panel for, I think it's for parentage assignment, but they have added this as it is quite an interesting QTL to add.
Okay, so a really last short question from C.
Yes, so this is a follow-up question of the QTL and industry applications, because there seems to be a number of studies finding the same QTL, and kind of a very attractive study would be to study this across populations. You think are there any initiatives to do that, do you think?
Yes, I think it is quite an interesting idea, and we believe the French team has got a very big population, and actually they are trying to validate SNP markers they identified, though we have quite better results. But yeah, they are trying to validate it, but I think yeah, the best idea from here going forward will be to merge and also see, because also I saw a similar study we shared last time from Norway. They also have a very huge population. So I think the best idea will be to merge these populations and see if we could validate the QTL in all the populations and have a refined kind of answer to the problem.
Yeah, super, really great. Thank you so much. So now I have the great pleasure to introduce our guest speaker, which is Dr. Wendy Bickmore. So she is a director of the MRC Human Genetic Unit. And she is really fascinated about the three-dimensional organization of the human genome, which is my case too, and how that influences genome functions in health and diseases. So yeah, thank you very much to join us today, and we are really pleased to have you.
Thank you very much. Thank you for inviting a human to your fish community. It's really, really nice to be meeting you, because we share a very common interest in understanding how the genome is regulated functionally, particularly from elements that are not actually within gene bodies themselves, but from outside of gene bodies. So I don't have to convince this audience, I don't think, that the complexity of vertebrates depends not on the number of genes that we contain in our genomes, but probably the number of regulatory elements or enhancers that are in our genomes. And I think it's quite, for the human genome, we've had that sequence for decades. I think it's embarrassing that we still don't know how many enhancers there are in the genome, where they are, what they regulate, how they do it, when they do it. So it's a massive hole in our understanding of genome function, I think. But of course, what enhancers allow you to do is to drive complexity using a limited set of genes, so that by having different enhancers regulating the same gene, you can specifically activate that gene, for example, to help pattern the eye during early development, to perhaps help specific neurons in the brain respond to particular stimulus, like learning, for example, or, for example, to pattern an appendage such as the limb in land vertebrates, or a fish fin, for example. So I'm going to try and tackle three particular questions that interest my lab in this talk. So it's kind of a flavour of the kind of things we're trying to do, to understand how enhancers work right from the basic level of individual nucleosomes on these regulatory regions, and then asking, and I'm essentially a cell biologist, I'm really interested in how enhancers work from ridiculous genomic distances from the genes they regulate, particularly in human genomes. And then I'm going to give you a flavour of, I think, an interesting direction we're trying to take this in using synthetic biology to try and understand regulatory landscapes. Okay, so let's start with histones. You guys are already using that in your functional annotation project. And as in fish and animal genomes, in human genomes, people have tended to use markers of chromatin modification, particularly H3K27 acetylation, to mark active regulatory regions. It works very nicely, it marks them, of course, and we think that these histone acetylations act as binding sites for effector proteins that then modulate some aspect of chromatin function. However, I think there was this very nice paper last year from Christian Helland's lab at the Institute of Cancer Research in London, where they mutated every single lysine 27 on histone H3 in cells, changed them to arginine, so they can't be acetylated. And you can see there's no K27 signal left there anymore. Oops, I'm missing the laser pointer there. There it is. See, it's pretty much gone. But surprisingly, they found almost no effect on gene expression. So does K27 acetylation actually matter? It's a nice biomarker, might not be very functional. They found a massive effect on polycomb repression by K27 methylation. That's clearly absolutely vital. Acetylation, really not clear what it's doing. But I think it would be appropriate for me to remind this audience that actually histone tails are not the only place you get post-translational modifications in chromatin. You can also acetylate histone H3 particularly within the globular domain, within the heart of the nucleosome particle. and particularly around this region of the nucleosome that's called the diad axis. This is really interesting part of the nucleosome because it's where there's only a single pass of the DNA across the histone optima. So anything that perturbs DNA-protein interactions in this region of the nucleosome could have very dramatic effects on nucleosome stability. And so several years ago now, we'd been working with Robert Schneider's group in Munich, who developed antibodies that detect modifications within the globular domain of histone H3. And we were, for example, able to show that acetylation of this lysine here, lysine 122, close to the diad axis, very much kind of matches what you would expect for active regulatory regions. It's present at a subset of H3K27 acetyl sites. And we were able to go on and show functionally by deleting some of these elements. And we found, for example, some elements here. Here's a K27 acetyl peak in a non-coding genome here that has no real significant sniff of H3K27 acetylation there. So we asked, is that functional? We deleted by CRISPR and showed that it was in between two genes here, and we showed that this gene here is affected by deletion of that element. So it suggests it's an active regulatory region you just wouldn't see by K27 acetylation. Okay, so moving on, we realized that there's another, as well as lysine 1222, so which sits here, very close to the diad axis here, very close to the overlying DNA, there's another interesting lysine on histone H3, which is histone 115 here. It's actually the closest lysine to the overlying DNA. And indeed, this lysine can get acetylated as well. And there are pretty decent antibodies for this modification. But nobody had really bothered to look to see where it is in the genome or what it might do. So we thought we'd take a punt at this. So this is the work of two postdocs in the lab, Adipta and Yatendra. So we generated chip sequencing data in mouse embryonic stem cells to start with, and we did some differentiation as well for lysine 115 acetylation. And you can see it gives you really nice signals at distal regulatory sites and over gene promoters as well. And it seems much more precise, I would say, than the K27 or even the 122 acetyl profile. So one thing in my lab we always do when we're doing chip sequencing is we always do native chip. We don't cross-link. You get better cross-linking to chromatin that way. And we sequence in paired end so that we can actually map exactly where the nucleosome sits. So you'll see in a minute why I think this is an important thing to do. It gives you extra power. So firstly, we're just going to focus now on transcriptional start sites for a bit. So you can see, so this is our native RNA sequencing, so TT sequencing. Basically, we put thio-UTP into these cells. So you can see that the level of 115 acetylation very nicely scales with transcriptional activity. And then when you look at the heat maps, you see something interesting. So here's the K27 acetylation around a transcription start site. What you would see, it's on the -1 and the +1 nucleosome relative to the start of transcription. 122, very similar, -1, +1 nucleosome. And you see that there in the overall profile. And when we looked at 115, we realized it looked really different. It wasn't biphasic like this. It was sitting right down the middle at position 0. But there's actually not supposed to be a nucleosome. So a little primer about what we think we know about nucleosome architecture around active promoters. So many of the textbooks would say promoters don't have nucleosomes. They're called nucleosome-depleted regions or nucleosome-free regions. But several groups have shown that actually there's a more nuanced situation than that. And so this diagram here is taken from one of Steve Henikoff's papers where he used much more limiting micrococcal nuclease digestion. And then in his chip sequencing, and again paired end sequencing, so you know the length of the fragment. And if you look at the canonical nucleosome size fragment, you indeed see there's nothing there at 0. So that would be consistent with your nucleosome-free region. But now if you look at your chip sequencing fragments that are a little bit shorter than what you expect for a fully protected nucleosome, you start to see something appearing at position 0. So it's thought that actually there is some kind of subnucleosome particle, fragile nucleosome, perhaps a hexamer, maybe a tetramer, no one's quite sure, actually sitting at the promoter. So it's not really nucleosome-free. So let's have a look at that in our data. So here's our data for K27 acetylation using this kind of approach. So we're plotting fragment size against distance from the transcription start site. So here's the input nucleosomes. So that's the -1, that's the +1, the +2. You can see there is a signal there from the MNAs there in the middle. And perhaps you can see that actually the signal extends lower than the canonical size of a nucleosome. For K27 acetylation, you really don't see anything at the position 0, and you see this massive signal at the +1 nucleosome. Now, what do we see when we look at our 115 data? Here we see what's really interesting. We see the signal's highly enriched right at 0, and in a particle that's slightly shorter than what you expect from a full nucleosome. So we think that this acetylation is very specifically marking, almost to nucleosome level, a very special nucleosome that sits at the start sites of highly active genes. My guess is it's partially unwrapped. It might be a hexosome. I think we need to do some electron microscopy to really understand what the nature of that is. So that was transcription start sites. What about enhancers? Is it there? And the answer is yes. And again, so we're going to do the same analysis here. Looking in mouse embryonic stem cells, we're going to focus on enhancers that are being regulated by the pluripotency factors, such as OCT4. And so we're going to center on the OCT4 motifs here. So this is the attack-seek signal at these kind of enhancers here. So you see that very nicely there. You almost see the footprint, actually, of the two motifs for OCT4 here. Here's the MNA's track. So here's the flanking nucleosomes around the active enhancer, and a massive dip in canonical micronuclease signal there, suggesting, again, there isn't a nucleosome there. Histone K27 acetylation does the same thing, depleted right over the binding sites for the transcription factors and enriched on the nucleosomes immediately flanking that. But when we look at 115, it's doing what it's doing at the transcription start site. It's sitting right where there's not supposed to be a nucleosome, right where the transcription factors are binding. Both actually in full nucleosome size fragments and also in subnucleosome size fragments. So we think 115 is very precisely marking where transcription factors are binding within regulatory elements. So it might give us a lot more precision in finding where the interesting part of an enhancer is compared to something like K27 acetylation. So K115 acetylation, not previously studied, seems to be a very powerful marker of active regulatory sites in the genome, both promoters and enhancers, probably marking partially unwrapped nucleosomes. Okay. So now the question, how do enhancers work at a distance? I confess I don't know enough about the fish genome to understand the size of regulatory regions in the fish genome. So I'm going to really focus on what we know about mammalian genomes. So we mainly work in the mouse and the human. Here's two of my, I like complexity. Here's two of my favorite loci. So they both encode genes really important in development. Sonic hedgehog, a major embryonic morphogen, PAX6, a major master transcription factor for things like the eye and the brain. And their regulatory landscapes are ridiculously complicated and large. And so this information has come from really decades of human and mouse genetics to define this. Here's sonic hedgehog here, tiny little gene over here, polycomb target in stem cells. And we know, I think, 18 enhancers so far that drive sonic hedgehog expression, different times and places during development. And they are scattered over almost 1 million base pairs of the genome upstream of sonic hedgehog, including in the introns of neighboring genes, including 2 genes away. This is the famous limb enhancer of sonic hedgehog, which is required to pattern the limb. PAX6 here, and again, pretty tiny gene. 34 different enhancers we know of that drive PAX6 expression at different times and places in development, scattered upstream of PAX6 in this non-coding region, and downstream for hundreds of kilobases of PAX6 within the intron of this housekeeping gene here, ELP4. So in fact, many GWAS for traits that involve PAX6 call the target gene ELP4. Genomically, that would make sense, but they're not. They're really PAX6 enhancers. So if I was to sit down and design a genome to regulate gene expression, I wouldn't do it like this. But this is what evolution cobbled together. So how on earth does it work? How can a bit of non-coding DNA stuck in the intron of a housekeeping gene so precisely regulate sonic hedgehog expression during development to restrict its expression to a tiny patch of mesenchyme on the distal posterior part of the hand plate that's going to dictate how many digits you have and what their identity is. So amazing precision, but from such a large genomic distance. So I think the progress in this field really came from the omic technologies that allow you to profile the 3D organization of the genome, which I'm not really going to go into, but they're basically around the 3C, high C family of technologies. So they're kind of ligation, proximity ligation-based assays. And they showed that at least our genomes, mammalian genomes, are organized into triangles, which we call topologically associated domains that you generate from these data, which basically mean all the sequences in here tend to interact, in quotation marks, or ligate together more often to each other than they do to the neighboring sequences over here, even though the linear genomic distance might be shorter. And when you look at these, they align so beautifully with regulatory domains. So here's the sonic hedgehog domain here. Here's sonic hedgehog over the left-hand side of this TAD, which is 1 million base pairs in size and contains all the known enhancers for sonic hedgehog, including that distal limb enhancer there. And you see that for many other loci as well. So this is kind of smoking gun that says there's something about TADs that must be important for enhancers to work. And just for those of you who don't know how TADs are made, they're made by an amazing process called cohesin-mediated loop extrusion. So cohesin is this ATPase-driven translocase, I would call it, that moves up and down our genome 10 times faster than any polymerase we know of. And as it does that, it kind of topologically traps chromatin, scrunches it up, until it hits a brick wall, which are very often CTCF sites in mammalian genomes with a particular orientation. So that's how these triangles get made. One thing we should remember is this is not really a structure that exists. This is an ensemble of data from lots of cells in a population. So where these structures are in the process of made and destroyed. So they're being made and destroyed all the time. Cohesin is stripped off the genome by a protein called WOPL. Okay, so how can we test this? Well, we thought we'd like to do that for the sonic hedgehog lockers and ask whether this TAD matters. So we just used classical mouse genetics. What we did was we just used CRISPR to take out the boundaries of this TAD. We deleted the CTCF sites. We did it homozygously. We did every CTCF site we could find in this region. There were 5 of them. Lots of work. Made mouse embryos from these mutant cells, and we found absolutely nothing. Sonic hedgehog expression was completely normal, expressed in all the right places, all the right times, and mice had no overt phenotype whatsoever. Even though, for example, when we deleted this boundary, we could show by 3C type technologies that indeed we'd lost this boundary and we created a new one here so that sonic hedgehog was no longer in the same TAD as enhancers. So that says at least the precise architecture of that triangle doesn't matter that much for gene expression. But that doesn't mean to say that the process of making the triangles doesn't matter. What about the cohesin-mediated loop extrusion, the active process itself rather than the final structure? Is that important? So how can we address that? Well, we can't do it by classical genetics because cohesin is an essential protein. You need it for cell division. So you've got to come up with a different way of doing that. So to do that, we've taken advantage of the Degron systems that have been developed for vertebrate cells and for other organisms as well, which are really powerful and allow you to remove the product of a gene very rapidly by the addition of small molecules. So we've used the auxin system, so where there's an auxin-induced Degron fused to the protein of interest. You add auxin, the plant hormone, to the cells, and within a few hours your protein is gone because it's taken to the proteasome for degradation. So here I'm showing you data from a system where we have the cohesin itself tagged with an auxin Degron. We got these cells from Rob Close's lab in Oxford. It works really well. All the cohesin that we can detect is gone within a few hours in the cell by the addition of auxin, and you can see that in the high C map. So if you look, this is the sonic hedgehog TAD now. I've turned it on its side, so it's a square now rather than a triangle here. When you add the auxin, it just melts away. There isn't a TAD there anymore. There's nothing actually creating the 3D genome in this region anymore. So that's good. So we can do that in an embryonic stem cell, so we can ablate that. But how can we ask, can these enhancers work now? Because these are tissue-specific enhancers. They don't work in stem cells. Sonic's off. So, and we can't use development because you need cohesin for development. So what do? So what we do here is we embrace synthetic biology and say, okay, we're not going to wait for differentiation or development to turn our enhancers on. We'll turn it on ourselves with synthetic transcription factors. So that's what we have done. Oh, I'm also meant to show you. You can really see the effect of losing cohesin visually as well, because a consequence of cohesin-mediated loop extrusion is that all the chromatin in a TAD is kept very compact. So if you label the sonic hedgehog TAD with 3 probes, one on the left-hand side, one on the right, and one in the middle, and remember this is 1 million base pairs of the genome, and you look in wild-type cells, you see all 3 probes really tightly together. But when you get rid of cohesin by adding AUX in, you can really see the whole structure relax. The whole volume of the domain goes up, which I think is the important factor. So one reason that people hadn't really pursued cohesin-mediated loop extrusion enhancer function before was because when people first discovered this mechanism and they used siRNAs and other things to deplete cohesin and then did RNA sequencing, they found nothing much happened. There wasn't really much change in gene expression if you just looked at regular RNA sequencing plus or minus auxin. But that's not the same as saying, can I activate a gene from an enhancer when there's no cohesin? Because this is steady-state RNA. You're not asking the cells to do anything. So instead, we decided to ask the cells to activate sonic hedgehog synthetically. So our synthetic transcription factors we designed are built on a TAL effector backbone, which we think is much better than a dead Cas9 backbone for an activator. Dead Cas9 is fine for repression. It's not a very good activator. I can discuss that later if you want. And we turn it into a transcription factor by putting in a multimer of VP16, a well-known activation domain from herpes simplex virus. And our readout is RNA fish. So we're actually measuring at the single allele level whether we can activate sonic hedgehog or not, remembering it's actually a polycomb target in these cells. It's got K27 methylation all over it, and it's silenced. But it works very well. Here's a synthetic transcription factor. We actually targeted right to the promoter of sonic hedgehog, and I hope you can see here we're able to activate sonic hedgehog. But what about, can we do this same trick from enhancers? Here we're targeting a synthetic transcription factor to this enhancer for sonic hedgehog. It's actually a 4-brain enhancer for sonic hedgehog. It's almost 500 kilobases away from the gene, but we can still turn sonic hedgehog on synthetically when we do that. And we can even do it from the limb enhancer of sonic hedgehog, 1 million base pairs away, stuck inside the intron of a housekeeping gene. And there's 2 little spots there of sonic hedgehog expression. So we've now got control over cohesin by adding auxin, and we've got control over enhancers by synthetic transcription factors. So we put the 2 things together, this was published last year, and asked, do enhancers still work when cohesin's not there? And the answer was yes, sometimes. So we could still activate sonic hedgehog at the promoter when you don't have cohesin, so it's not as if you need cohesin-mediated loop extrusion for transcriptional activation per se. We can still activate from this enhancer of sonic hedgehog, 100 kilobases away from the gene, but we could no longer activate from these more long-range enhancers. So it looks like we need cohesin-mediated loop extrusion, the change in the 3D genome that brings about to allow linearly far-away enhancers to work on their target genes. So this allows vertebrate genomes to really develop these really long regulatory regions. So the way we're actually thinking about how enhancers work is, and I'm not going to go into the background of are within the sphere of influence of the signal that's coming out of the enhancer. That diffusible signal, whatever it is, can reach the target gene. But when you degrade cohesin, for an enhancer that's close to that promoter, it's still within that kind of sphere of influence, that radius, because it's not very far away in the genome. So it can still work. Whereas a distal enhancer, 1 million base pairs away, once you take cohesin away, everything decompacts. That gene can no longer receive or hear or respond to the signal coming out of the enhancer because it's just too far away in the nucleus. Size of that sphere of influence, we're not sure. It's of the order probably of 200 to 300 nanometers. That's what we're thinking of in the field. And obviously we want to understand more about what the signal is and exactly the size of this region and whether it's different for different enhancers, for example. Okay, so that's how we think enhancers work from a distance. Now to the question of can we kind of get our head around why some of these regulatory landscapes are so complicated. So now I'm going to focus in not on the sonic hedgehog locus, but on our other favourite region, the PAC6 locus here, just to remind you what it looks like. Here's PAC6, there's enhancers in the introns of the gene, in the introns of this downstream gene, and upstream of this gene. And if you look at how these enhancers are annotated, it's pretty crude, and there's a whole bunch of different enhancers that are just labelled as neural retina. PAC6 is really important. It's a master regulator of eye development. So the question is, well, so do all these different neuroretinal enhancers do different things, or is there redundancy, for example, in the system, as is proposed in Drosophila, where there's things called shadow enhancers, kind of backup enhancers? To answer this, we wanted to really go in and understand these enhancers in more detail. So how can we do this? I do not believe you can understand enhancers such as this using cell lines. I think it has to be done in vivo, because we do not have cell lines that represent the right cell types at the right stage of development. So you've got to go into in vivo biology and do in vivo testing, and we've chosen zebrafish as our model system for doing testing much faster than the mouse. And of course, transparent embryos allow us to use imaging to study enhancer activity as well. So this is the work of a senior postdoctoral fellow in my lab, Shipra Bhatia. She developed this whole system. It was her idea. So she developed this beautiful transgenic reporter system called QSTAR, in which we can pit two different enhancers against each other in the same cells of the same fish at the same time and over time. The construct basically has one enhancer driving cherry, the other driving GFP, and we built a really strong insulator between the two that we've shown stops any crosstalk between the enhancers. And we can swap the colours around. We can have one enhancer driving the other enhancer driving cherry and the other enhancer driving GFP. And we put it into the fish either through a specific safe landing site with 5C31 or just using TOL2. And we get very similar results, actually. So here we're going to ask, actually, so these two neuroretinal enhancers, HS5, which is 167 kilobases downstream of PAC6 in the intron of ELK4, or NRE, which is stuck in the middle of an intron of PAC6, do they do similar things? And the answer is clearly no, using the QSTAR assay. So here you're seeing HS5 driving cherry. It's expressed in the eye, as we would expect, in the retina. But it's also expressed in a part of the forebrain of the fish and a part of the neural tubes. It's got other activities as well, whereas NRE is purely neuroretina. You can see it beautifully decorating the eye. If we swap the colours around, you see the same thing. Okay? And what we found was very interesting was that HS5 in the neuroretina was very different from NRE in that it was restricted to this temporal lobe of the retina. It wasn't all over the retina. So there's already, within an early stage of a developing tissue, a spatial difference in the activity of these enhancers. So we can follow, because this is live cell imaging, we can follow how this develops over developmental time. So this is 24 hours. This is what you've seen already, with the HS5 activity restricted to this temporal lobe. By the time you get to 48 hours of zebrafish development, you can still see restriction to the temporal lobe here and the ventral part of the eye. And actually now you can see the two activities separating out into the inner and outer retina as well. And really by 72 hours, where you've got different cell type specification going on in the retina, you can really see that the two enhancers are really driving activity in different cell types within the retina. So our conclusion from this is these are not redundant enhancers. They have different functions in the eye and different activities. And you've really got to dig down deep into development and space to really understand what they are. So I think in terms of functional annotation of our genomes, we've got to go to this level of detail. And we can do a certain amount of it just by imaging, but we thought that wasn't good enough to really understand what those cells are. So Shipra and a student in the lab, Kirsty Utley, decided to couple this to single-cell RNA sequencing. There's a fantastic single-cell atlas of the developing zebrafish. So we got the lab together one day, lots of scalpels, and dissected loads of eyes from the QSTAR fish, and sent them for single-cell RNA sequencing, sorting for GFP and cherry, and then mapping that data back onto the single-cell atlas to see what was the nature of the HS5 and the NRE active cell types. And that was really interesting and worked really, really well and showed us, really gave us our first real insight into what these enhancers do. So NRE was active specifically in the stem cells of the neural retina at the ciliary marginal zone. You can see that there actually from this view of the eye. And then later on it became restricted to the amacrine cells, which are interneurons that talk to the retinal ganglia cells. Whereas HS5 was active just inside the stem cell zone of the retina in the first progenitors of the retina, and then also in the Muller glia, which are glial cells, one of the few glial cells in the eye that actually originate from the neuroretina. So we can really start to be able to see exactly what these enhancers do. We've got 32 other enhancers to work through for PAC6 before I think we really understand this. So that's some work to do. Okay, and now the bigger picture. Does it really matter that the regulatory landscape of PAC6 looks like this, or is it just an accident of evolution? Would it matter if all these enhancers over here were over here, and if these enhancers were over here? And does it matter that the spacing between those two is that like that? You know, if we put them together, would it matter? So, I mean, you couldn't do this by just individually CRISPRing and loxing and inverting individual bits of the genome. You have to think differently if you're going to ask those kinds of questions. So for this, we teamed up with Jeff Boke's lab in the USA. Jeff runs the Dark Matter project, and Jeff was the lead scientist for the Saccharomyces cerevisiae 2 genome, in which they completely redesigned and rewrote the whole of the genome of budding yeast. Amazing technical feat. So they've got the technology to do this, and they're really interested in expanding this now to tackle big genomes, vertebrate genomes. So they can do the rewriting of the genome, but they have no way of functionally assaying what comes out the other end. So we teamed up together to do this, funded by BBSRC in the UK and NSF in New York. And so the question we want to ask is, here's PAC6. If we put a reporter in so we can follow the activity of the enhancers acting on PAC6, what happens if we shrink the locus, if we bring all the enhancers together, or if we move them around? So we can rewrite now all these versions of the PAC6 locus. So that's ongoing. So Brianna, a student in Jeff's lab, is doing the building for us. We're still trying to do the mouse and human locus. It's 450 kilobases. It's the biggest thing they've ever tried to rewrite. And we can't move it into, for example, mammalian mouse embryonic stem cells as a oner. It's too big. So we're having to break it up, and it's a bit more complicated engineering. So we thought, okay, let's use a genome in evolution that's much smaller than the vertebrate genome and see if we can start to explore this. So we chose fugu, the pufferfish, which you probably all know very well. It's got a very contracted genome, but very similar to the vertebrate genome. So here's the 450 kilobase human PAC6 locus. Here's the fugu locus. It's only 70 kilobases in size. It's really small. And yet many of the enhancers that we recognize in the mammalian genome are there for PAC6, and they're all in exactly the same place. So that position has been conserved. So Brianna wrote us an artificial fugu PAC6 locus tagged with MKate downstream of PAC6. And Shipra and her colleagues delivered that synthetic genome to the zebrafish genome to ask, does it work? And the answer is yes, it's beautiful. So this is a completely synthetic bit of DNA we've designed, reporting on PAC6 expression through this tag here, and it's really showing us expression or activity in all the parts of the developing fish we would expect PAC6 to be expressed in. With some exceptions, we can't find the pancreas expression, so there's a few things that aren't quite working. But I think it's a good enough platform for us to start rearranging it. We've now got a fish where we've got a GFP tag here, so the same synthetic genome with the separate tag, and we've crossed our two fish together to show that they've got completely overlapping patterns of expression. So I think now we have the control fish. Now we're waiting for delivery of the fish where all these guys over here go over here and all these guys go over there to see what happens. I don't know what will happen, whether it'll matter or not, but it's going to be fun to find out. So that's the kind of thing we're doing. I think this is a really exciting area of functional biology, actually, for the future, to be able to rewrite things. And actually very well placed in Edinburgh to do that, because we've got the Genome Foundry at King's Buildings that can help build these kinds of complex constructs. So I hope I've shown you that acetylation on the lateral surface of histone H3 is a really important chromatin mark that's underused, I think, by the community. And it very precisely marks active regulatory elements and precisely where transcription factors are binding. So it's almost like a footprinting tool. Enhancers located distant, hundreds of kilobases from their target gene, require cohesin-mediated loop extrusion to work. And I think synthetic biology is a really powerful tool now that we can all use to understand complex genomes. So I'm going to end by thanking the people who did the lab in the work that I described here, which was Alba, Sheila, Yatendra, Elias, Shipra, Laura, Ian, Grace. Thanks to Rob Close for his cohesin degron cells, and to Jeff and Brianna for the synthetic biology. And big thanks to the facilities at the IGC, Lee Murphy, who does at the clinical research facility, who does our genome sequencing for us. He's great. Our imaging facility, our FAANG and single cell facility, and of course our zebrafish facility. And thank you for listening.
It's amazing. Thank you so much for this share about how long distance communication also is really important for regulation. It's what I try to implement in Norway. Boris.
Great talk, thank you. So first, about the first part of the talk,
yes,
about these nucleosomes that are phantom nucleosomes, that basically you see them in nucleosome-free regions.
Yes.
First, I was interested what kind of TSS definition you used as a reference point.
Oh, that's a good question. I don't know. I'd have to ask you, Tendra, but it does. It's the same definition gives you that gap down the middle of K27 acetylation.
So if you use like cage data, for example, the dominant peak, you have single base pair resolution, and it's aligns very well with the first downstream nucleosome.
Right. No,
I'm just wondering, if that nucleosome that you see, if it also aligns that well with the actual transcription.
We should do that. We haven't used cage data. We've just used annotations from
and
UCSC, basically.
The second thing about your basically last slide. So you said when you express the tetraodon PAC6 locus in zebrafish.
Yes.
You don't see the pancreas expression.
No.
Have you looked at its PAC6A locus, because this is PAC6B, right?
Well, no, this is this.
Yes.
Yes, it is. Yes, yeah. But the element that in mouse is annotated as the pancreas enhancer is present in our construct. So we were expecting to see it, but we don't.
Right. Okay. Yeah. Okay. Thank you.
Yeah.
And I think for our question of whether we mess around with your order, it doesn't actually matter. We're not asking about every single answer. We're asking big questions about the overall architecture. Yeah.
They are collinear mostly, right?
They are. Yeah.
See.
Thanks a lot. Very, very interesting talk, and very fascinating. All this, the methods you have to play and to understand regulatory machinery to regulate systems. So in AQUA-FAANG we have taken as we are studying different species. So in a way, the target there is to study this in different species and maybe look for natural differences between species and try to understand this machinery by comparative genomics. Yeah. Maybe you could comment a bit on how realistic you think that will be?
Yeah, what are the right tools to do this? I mean, you guys are using K27 as a TAC-seq and RNAs. It all makes sense. And people in the human GWAS field are doing exactly the same thing. We have a study not quite ready for publication yet, where we've gone through commonly used regulatory marks. We haven't used 115 acetylation yet because we haven't got enough cell type data. To ask what gives the most precision in terms of linking GWAS SNPs to causal variants in regulatory sites. And in fact, a TAC-seq and K27 selection give great recall but very poor precision. In fact, we found the thing that gives the greatest precision was a combination of using eRNAs, so short bidirectional transcription, coupled to DNA's hypersensitive site footprints. So again, much more precise than the TAC-seq peaks. So I'm not quite ready to say to you guys, actually, perhaps you should think about in your next step, thinking about things like that. I do think bidirectional eRNA is an incredibly powerful, actually, way to do it. How much you can do that at single cell level is unclear because, of course, they're not very stable. You have to sequence quite deep to see them in regular nascent RNA sequencing, for example. But I think the coupling of those two, I think, might be the way to go.
Thank you.
Hello. Here. Thank you for the wonderful talk. So I have a couple of questions regarding the first two parts, so the first and the second. So in the first part, do you have any evidence maybe that this possible nucleosome interact with either the pre-initiation complex on the promoter or...
Yeah, that's a really good question. We don't have any evidence yet. We have evidence that it's important for transcription. So again, this is a collaboration with Robert Schneider's lab in Munich. He's an expert on in vitro transcription on chromatinized templates. And so he can make chromatin with nucleosomes decorated with different modifications and ask if you just add the transcriptional machinery, how well does it work? And there, you add K27 acetyl and nothing happens. There's no readers, nothing. But all these globular domain acetylations boost transcription in vitro without any readers or anything else there, which we interpret probably as the fact that it destabilizes nucleosomes, it makes it more efficient for the transcriptional machinery comes in. So it might be as simple as that. But it could be a direct interaction, but we've got no evidence for that just now. I think it would be hard to think of how a component of the transcription machinery would read something that's inside the nucleosome particle, but that's not without possibilities. I think we'd need some high-resolution cryo-EM. And in fact, the people who've done the cryo-EM so far on the transcriptional machinery, which is beautiful work, have not included nucleosomes much in their work. Or if they have, they put the nucleosomes at +1 and nothing at 0.
Yep, and for the second question about the second part of the talk, like when you observe that the loop extrusion actually influences like real long-range interaction, have you seen what maybe if there is any polycomb influence on that, and if there is some chromatin remodeling process?
That's a very good question because the genes we look at are polycomb targets. So the enhancers are overcoming polycomb repression, and that's a question we are currently exploring because we're seeing some enhancers that act on sonic hedgehog able to act, we think, as a bystander on another gene in the other direction, another TAD that happens to be a polycomb site as well. And we know that polycomb sites in the genome stick together like glue in 3D space, so they're very close to each other. So it's a really interesting question. I don't have the answer, but it's something we're actively exploring. Yeah.
Okay, thank you.
Okay, so, yeah, maybe a last question, yes? Yes. Yeah, we have time. We will have the coffee break after. So.
No, really fascinating research, thank you. My question's about TADs, which is, I know nothing about them, so how widespread through multicellular organisms are they? And is that linked to complexity of the animal itself?
Yes, it's a bit of a complicated answer. So yes, it's restricted to complex metazoans, not there in C. elegans as far as I know. Drosophila has TADs, but they look like they're formed by a very different process. So I don't think they're functionally equivalent to vertebrate TADs. But as far as I know, all vertebrate genomes have TADs. I don't know enough about fish genomes and what their average size is and what they look like. I don't know if anybody's really done that. I don't know if anyone in the audience knows how much high C data has been done for fish.
There's good zebrafish.
There is, yeah. And what's the average size of a TAD in zebrafish?
It shrinks the same way.
It scales with the genome, yeah.
I mean, we showed that this developmental tasks they coincide with this cluster of conservation, right?
Yeah.
And basically they also do with
right.
zebrafish.
Yeah.
So and when you have like a PAC6 locus that shrunk to 70 kilobases, then the TAD
shrinks accordingly, yeah. So I'm pretty sure in the fish species you look at, this mechanism is also at play. I think it's common to all vertebrates.
Any other question? No? So thank you, Dr. Bigvor, for this amazing talk. So now I can announce that we have a coffee break, and it will be until 20 past. So enjoy.
So welcome back to the third session of AQUA-FAANG. First of all, I want to remind to the people that are watching the online AQUA-FAANG conference that, yeah. So I want to remind to the people that are watching the online, the streaming version of the conference that they can ask questions, so whenever they want. So our next speaker is Lukas Napora-Rutkowski from the Polish Institute. And he will talk about the transcriptional differences cyprinid herpes virus-free response between resistant and susceptible common carp crossings.
Okay.
The floor is yours.
Yeah, thank you. Good morning, everyone. My name is Łukasz Napora-Rutkowski, and I work in Polish Academy of Sciences, Institute of Ichthyobiology and Aquaculture in Gołysz, in Poland. I want to thank organizers of this conference and AQUA-FAANG's leaders for inviting me and letting me present the results of our research about transcriptional differences in cyprinus herpes virus-free response between resistant and susceptible common carp crossings. So, cyprinus herpes virus-free is also known as koi herpes virus, and it's double-stranded virus member of Alloherpes virida. It was discovered and described in 1998, when it caused mass mortalities in Israel, in the United States, and Germany of common carps. It caused mortalities of koi carp, of common carp breeding lines, but also wild populations. It's distributed all over the world, except Australia. So our world is fighting against, especially people working with carp, fight against koi herpes virus, except Australia that is thinking about importing this virus to the continent to fight using this virus against the common carp that is invasive species at this continent. In 2018, we started a family selection program that the main goal was to produce, that is the main goal to produce the line of common carp that will be resistant, highly resistant to koi herpes virus. And we used Amur common carp, that is this one, that have high resistance to this virus, but it's a slow-growing fish with full scale. In our region of Europe, we like carp without scales and also like growing fast, what is good for aquaculture. So, but the resistance of these breeding lines, except this, that in Gołysz we have, because Gołysz also work as a gene living bank, and we protect, we have 16 different lines, breeding lines of common carp, this collection from all over the world, and also from, and there are also like traditional Polish common carp lines. And for this selection program, we choose to cross a few of these lines with Amur common carp, and we produce 16 crossings family. These families were produced in single crossing, like there were single parents for each family, so one male, one female. It was not mass spawning. So we produced 16 such families. We were using our facility of recirculation systems, especially for experiments, but we also have this on earthen ponds. like the same families are in both systems. So to check if our crossing families are really resistant, we had to we perform a lot of challenge tests. This challenge test, we use the system of infection by cohabitation. So we were infecting few fish that are really susceptible to this virus and putting them together into tank with the fish of the family that we are testing. So after multiple trials, we tested this 16 crossing family, these first generation families, and we observed that the mean survival of these families was from 21% to 73%. So after we finished this with this first generation families, we produced 30 backcross families. These backcrossings we were doing by choosing 6 best F1 families and crossing them back to the mother family of the founder. So we were crossing this to this line that gives the establishment of this family, this F1 family. And this is how it's like a ground fish. In fact, it's a small breeder, but more or less how this fish looks like. So it looks almost like agriculture carp that producers expect. So we tested these 30 backcross families one more time to check, to establish the level of resistance. In this presentation, I use, for me, in this situation, like survival and resistance is almost the same. Like for me, resistance carp is this one that survived. Its resistance is the level of fish, amount of the fish that survived our challenge test. So we analyzed these 30 backcross families, and for challenge experiment and for RNA-seq experiment to analyze these differentially expressed genes, we choose 2 most resistant families and 2 most susceptible families. So in this, and we perform challenge experiments like we did all the time. The only reason was this, that this time we were sampling fish for RNA-seq analysis. This percent is survivality of this challenge test. So it was good experiment because the resistance family was 82% survived, and 66% survived. The susceptible families were very low survivality. So we were sampling during these experiments 2 times the fish. So it was just before starting the infection. It is day 0, and it was like 1 or 2 days just before the mass mortality started. Because KHV infections, it looks like that the virus developed for around 10 days, and then by 2 or 3 days, all fish die. So just before this mass mortality start, we choose the point of sampling fish for RNA-seq analysis. We sampled 8 fish at every time point for 3 organs. It was head, kidney, liver, and spleen. After the experiment finished, after we isolated the RNAs, we isolated full RNA for RNA-seq analysis and send it for RNA-seq sequencing. And after we get this data, the differential expression analysis between this resistant and susceptible families was performed, and also the 2 functional analyses were performed. It was overrepresentation analysis and gene enrichment analysis. for this gene expression and this functional analysis, we were, in fact, we analyzed 4 different comparisons. Here I will be talking about two most interesting that I think. So there will be different expression analysis between resistance curves against susceptible at the beginning of experiment, so day 0, and resistance against susceptible at 11 day of the infection. The differential expression analysis gave us thousands of genes upregulated and downregulated, what was expected, and this what we get is the functional analysis indicated us that in fact head, kidney, and spleen tissue behave very similar at the beginning of experiment, day 0, at the 11th day of experiment. So there were very similar patterns of expression, especially connected with immunology genes. So this resistant fish, they had significant downregulations of genes connected with wound healing, blood coagulation, response to wounding. So here we see enrichment of head kidney. So here is this gene sets for this blood coagulation pathways. And here are these gene sets for spleen analysis. All of them are downregulated. And also this immunological gene, especially connected with complement activation, humoral immunoresponse. Here is the pathways of head kidney, and here are the pathways of spleen enrichment. And these dots, they show the p-value. All of these p-value are significant. So for me, it is interesting that these resistance lines, these high resistance lines, they have downregulations of these genes that are involved in immunoanswer. After 11 days of infections, of challenge experiment, also the head, kidney, and spleen showed similar results. So, but this times it's changed that there was upregulations of antigen processings and immunoresponse genes in head, kidney, and in spleen. But still, the complement activation in spleen and in kidney was down-regulated in these resistance lines. The genes of antigen processing were significantly upregulated. In kidney, it was also observed that the cell adhesion pathways are upregulated. Liver functional analysis showed us the upregulations of metabolic processes of lipoprotein and lipid transport. So generally, like metabolic processes were upregulated, and also the expression processes like translation, tRNA transportation was upregulated at day 0, and at day 11. Still, the immunoanswer of this antigen processing and presentation was upregulated. So this is of course just like first part of this research. Now we will be looking into genes to see exactly which of them take the most part in this answer, and we try to connect them with other data. But to conclude this beginning of this research, of this analysis, in fact, I would conclude it like that, this functionalized analysis indicated that antigen processing and presentation processes on cell surface are associated with MHC protein complex might be important in establishing this higher resistance of for this virus in these resistance lines, or higher resistance lines. And also it's indicate that this complement system might function differently in resistant line than susceptible line. So I think it also might be connected with this higher resistance. What was also indicated by a group from China who works on KHV, that they also indicated that this complement system works different in this resistance lines, but they use not amur carb but red carb in their selection program. And also like more business aspect of this project is this, that we have the 6 families, 6 highly resistant families with resistance of more than 60% that we bred in our institute. Last year we start to use them for producing hatchlings, because we also produce hatchlings. And we use the spawners to cross them also with these breeding lines and to provide hatchlings for aquaculture. So thank you very much for attention. I would like to thank you all and AQUA-FAANG for allowing us to do this research and to give us so much courses and cooperation. That was very nice, and I hope it will last further. Thank you very much.
Thank you, Lucas. Are there any questions? Okay, I have one. So you say that you are breeding strain, common carp strain, that are resistant to cyprinid-herpes virus. So you started a selective breeding program. Okay, so it's what kind of selective breeding programs you're doing? So it's
abstractive.
Sorry?
Abstractive.
So my question is it a traditional selective breeding program, or you are using genomics? What you're
now, in fact, we can say it was traditional, because these are the first genetic tools that we use for this program. Okay. So further, we will use them, and then we will be able to tell that it's genomic selection. But now it was, in fact, traditional family selection program.
Okay. No, it's really good. I mean, it's a very good results anyway. So, questions? Yes.
Thanks very much. It was an interesting talk. You mentioned in your conclusions that you felt that MHC potentially could have been a strong driver of your selection. Have you genotyped these fish for just the MHC alleles that they have, or were you basing it mainly on the fact that you've got MHC pathway presentation genes being upregulated in your RNA-seq?
I didn't sequence yet. These MHC genes in common carp, they were always popular to analyze in this KHV study. But here I just assume, because they are strongly upregulated. But of course, my next step will be to see the polymorphism of these genes, to see if in amur there is really different alleles, or it's somewhere else. Because in fact, there was many studies in this KHV and breeding lines and MHC genes. There were some results also indicated some positive value of some alleles, but it was just a few percent. It was 5% or something. But it was based on these breeding lines, not on the, let's say, wild carp, because we cannot say it's wild because it was from institute in Czech Republic, but it's supposed to be kept as wild. So I haven't checked yet the polymorphism, but I'm sure I will do it.
Okay, thanks very much.
Thank you.
Other questions? Okay, so thank you, Lucas.
Thank you.
The next speaker is Hendrik Jan Megens from Wageningen University, and he will talk about symmetric expression of analogs encoding conserved antiviral responses in common carp.
Okay, can you hear me fine? Good. So, my name is Hendrik Jan Megens, and yeah, I actually shouldn't be here. Well, I should be here, but not here presenting because actually Annemiek was supposed to present her work today, but she's enjoying a nice vacation. She couldn't make it, but she wants to sort of say hi to everybody. I also would like to include Mark here. Mark has been very instrumental in all the molecular work during COVID, mostly while juggling his own PhD project, which is unrelated. So very nice that he's been able to do that. And Geert also couldn't be here today. Of course, he's been from the start instrumental to the Wageningen contribution in AQUA-FAANG. So I hope I will do justice to Annemiek's work. Let's see how that goes. So one of the things that yesterday already was quite apparent in some of the talks is that we are blessed in AQUA-FAANG to have no fewer than 3 out of 6 species that are in fact tetraploids. Tetraploids are quite interesting because what will happen to a polyploid genome over time is that the genome will try to sort of become diploid again, rediploidization, which entails gene loss. That seems quite logical. I mean, you have genes redundant, so why not? But there may be all sorts of issues with things like dosage compensation. Genes may also be retained, but then acquire specialized functions. So this is, from a genome perspective, it's quite an interesting sort of feature to study in genomes. In carp, we have a so-called allotetraploid, so that means that we actually have the product of a hybridization of 2 species. And since these genomes started out different, over time there has been no recombination. Another difference with the salmonids is that it's recent, well it's recent, 12 million years, 13 million years, that this has happened, as opposed to the salmonids which like 80 or 100 million years ago when their autotetraploid event happened. So yesterday was also remarked that it's actually a very nice opportunity because you have 2 of these genomes that are traveling essentially for 12 million years in the same organism being exposed to the same conditions. So this is really very nice, so there is no recombination. At the same time, because this has been quite recent, the genomes are still quite similar, and that in itself poses problems when you have to study processes like gene loss, because if you assemble a genome, yeah, you will actually sort of compress regions that are very similar. So if like certain regions with genes are too similar, you might actually sort of assemble them into one. So how are you going to actually confirm that you have bona fide gene loss? And in addition, one of the things that you would hope to do is making full use of the fact that you have these 2 separate genomes, sort them by ancestry. And ideally, you would do that on a chromosome level. So when we started out this research, there was no genome available that was actually sort of allowing us to do exactly that. So we started out building a new genome with nanopore long reads, Illumina for polishing an IC to make a contact map, which this is just a few years ago, actually seeing where we are currently with telomere to telomere genomes being the norm almost. It's quite mind-boggling to see how fast things are moving. But yeah, so we were actually able to create a chromosome level assembly based on high C scaffolding. This is the results. You can actually very nicely see 50 chromosomes assembled. The genome is now available through Ensemble, has been annotated by EBI. So that's a huge resource for us to use. In addition, from this contact map, maybe also referring to one of the previous speakers, one of the things that we actually don't see in this contact map is any evidence for, let's say, the homologous pairs, chromosome pairs, to actually show evidence of interactions, which I think is quite interesting in itself. This is just one sample from blood, but there is really no evidence of that. So what Annemiek did subsequently is take the genome of this Poropuntius species, which is a model for one of the ancestors, and use that to sort of create a map of similarities, sort of allowing, following on the footsteps of Su et al. From 2019 to really sort of disentangle the genome into two. And so what we ended up with is this rather nice resolved sort of binning of genomes into CARP subgenome A and CARP subgenome B. Ada yesterday already mentioned this. And those chromosomes are not only synthetic to each other, but also completely synthetic to zebrafish. So the 25 zebrafish chromosomes have a duplicate representation, in a sense, in the CARP genome. So this is a very nice resource to start working on our basic problems. So then the next question is, do we actually have a complete gene set? So one of the things that you can then do is do this so-called Busco analysis, where you take a set of vertebrate genes, in this case, that we expect to be present in all vertebrates. These are genes that are selected because they have very strong one-to-one orthology in the vertebrates. And indeed, what we see is that the CARP genome seems to be, that we built, is pretty complete. What we wouldn't expect in a sort of normal diploid genome is high degree of duplicated genes, but they're there, and of course this makes sense. But then if we look at the subgenomes, we really see that those subgenomes really each are not as complete, which certainly suggests that both of the subgenomes are necessary for a functioning organism. And in addition, subgenome A seems to be a little bit less complete compared to subgenome B, which is interesting because subgenome A overall, as Ada yesterday also already mentioned, is less expressed on average than subgenome B. So there is a hint of directional gene loss, directional towards subgenome A. So this is something that we really wanted to follow up. And this is where context really starts mattering, I think. So yesterday Ada mentioned that as you go in ontogeny, the differences in between subgenome A and B in expression diminish. They start becoming less. And so it really seems that as certain conditions change, the sort of contribution of each of the subgenome may in fact also change. So one of the things that we ask ourselves here is, what happens if you really are inducing gene expression? And in the context of our experiment, this was directed specifically at the ISGs. Thomas yesterday already mentioned a lot about ISGs. And we used the same approach, essentially, but then really selecting the most conserved ISGs that have a very high one-to-one orthology relationship within fish and even invertebrates. And so then we end up with around 100 genes of different that have different different types of genes, transcription factors, enzymes, viral sensors, but all genes induced by interferon. So in zebrafish, there's about 100 of those. And looking at carp, Annemiek was able to actually find 91 back. And this was a painstaking analysis where she really had to sort of manually curate all these genes because the automated annotation from Ensemble was, was lacking here and there. This is, I think, expected. So it really requires very stringent manual curation. But after every search that you could think of, there were still 9 missing in subgenome A and 6 in subgenome B. So this really also indicates that also for this particular response, interferon-induced response, you really need to have both subgenomes. So we wanted to dissect this further and look at gene expression. And here, well, I actually will reiterate some of the aspects that were of the experiment that were already mentioned. This is actually one of the nice things, I think, within our consortium. For this particular experiment, it was done the same for all 6 species, which is actually quite special because normally if you have live agents and you want to do an actual challenge test on live agents, yeah, that's actually very difficult to do across species because all of these viruses will be different. With this strategy, we actually have quite a uniform experiment across 6 different species. So this will be recognizable. Several figures like this have been shown this morning. And what we actually turned out doing is focus on the in vitro experiment because that was giving us the clearest signal. Also talks earlier this morning already indicated that there may be some differences between what the in vivo and the in vitro experiments are telling us. So in the case of the carp, actually the in vitro was most sort of pronounced in its response and most easy to explain. Now this figure I don't expect you to study, but what I will just highlight is that we have these 100 or odd genes here that are color-coded similar to the figure earlier. And then these are the upregulations per subgenome. And what you should take home if you study this for a while is that actually there is no difference, no systematic difference between those subgenomes. So this is, in that sense, a bit of an uninformative figure because that is what the genome is telling us. Same thing if we look at the normalized counts in the controls versus the poly-IC stimulated condition, we actually see that there is upregulation, but it's not really very biased towards one of the genomes, and I can show that a little bit different in this figure, where if you look at the controls, the constituent expression is actually a little bit higher in subgenome A, which would be unexpected, but then after induction, after treatment, subgenome B is ever so slightly higher induced. But also, I mean, from a statistical perspective, not really significant. From a biological perspective, well, we can debate that. And that, of course, is something for further investigation. This is a different perspective. Looking at all upregulated genes, we actually see that there's, for each of the subgenomes, 600 odd genes that are upregulated. There is a significant overlap between the subgenomes for this experiment where genes are upregulated. But there's also a bit of a sort of a bias in one or the other subgenome, so this is all upregulated genes. Whereas if we just focus on the ISGs, we really see that those genes that we expect to be upregulated, most of them actually will, independent of the subgenome. Although there are a few genes that actually resist our expectation. And that, of course, is of some interest that we haven't actually followed up yet. So, briefly, conclusions. So we have mostly, in both of the subgenomes, we have this ISC gene set complete. And analogs are both expressed from both subgenomes during an immune response. But there is not really evidence for pronounced subgenome dominance. So again, here I think this context really matters again. If you want to learn more about this, actually there's a pre-release of this paper in Genomics just out for your perusal. So if you're interested in this, I would actually invite you to look it up on the internet. And then a last word about the future. Annemiek will come back in 2 weeks' time from holidays. And there's still a lot of work to do. One of the problems that we've had with this particular experiment is that actually our chip data didn't really sort of perform to our expectations. In the end, it turned out that we chose well, in retrospect, we chose the wrong kit. What can you do? But the ATAC is currently being very actively analyzed by Annemiek. Too early yet for conclusions, but this is certainly something where we want to study further the genome architecture, the regulatory landscape, let's say, of these differences, and then see why you actually have some similarities between subgenomes, but also differences. And I think Damir yesterday already sort of highlighted some of the potential sort of differences that we might expect there. And with that, I just want to thank all our friends in the carp genetics community and the fish immunology community that have been instrumental to this work. So, yeah, thank you.
Thank you very much. Questions? Yes.
Thanks, Hendrik Jan. You focused on the poly-IC in vitro because you said it gave the best sort of data. I was wondering, did the results hold up in vivo? And do you, because obviously you're hitting the cells quite hard and maybe encouraging the genome to be activated. Do the results hold up in vivo? Because I'm wondering if the in vitro might be like a over-exaggerated response of the subgenomes.
Yeah. That's a good question. And so we've had a good look at in vivo, in vitro. What we see generally, and the same for the Vibrio experiment, our in vivo experiment just it didn't really elicit a very strong response. So if you look at all the genes and the expectations of which genes should be upregulated, some things are there, but not quite. And probably, well, I'm not a fish immunology, but Geert is both carp biologist and immunologist. What we think is happening there is that it's a timing thing. So it might have been, prudent for this particular experiment to just wait for another day or something like that for the response to emerge. So we see something's happening, but it's just not as pronounced. And it's also more difficult to explain as a result, because we actually know quite well what should happen there. And it seems to be just in the beginnings.
Congrats on the paper.
Other questions? Okay, so thank you very much.
Okay.
And let's welcome our next speaker, Shamir Nazer from the University of Aberdeen. We will talk about genomic regulatory landscape underlying the antiviral response in Atlantic salmon.
Hello everyone, I'm Shamir Nasir. I'm a PhD student in Sam Martin's lab in Aberdeen. Today I'm going to present our work on some bits of our work package 3 work that we want to look at the multiomic data that we had from ATAC and CHIP to look at the regulatory regions and align the antiviral response in Atlantic salmon. I'm going to repeat the experimental design again for work package 3, which I think it was already done by 3 people today already. We had both in vivo and in vitro approaches where we injected 6 fish with poly-IC and 6 PVS control and kept them for 24 hours to get the innate immune responses and then harvested head kidney. For the in vitro, we first extracted primary leukocytes and then stimulated those primary leukocytes in 6 well plates and then made these assays, extracted RNA for RNA-seq and then made ATAC and CHIP libraries. We only made 2 CHIP libraries. We used H3K27AC as an active mark and H3K27Me3 as a repressor mark. So after sequencing, we identified around 53,000 unique ATAC reads and 72,000 the active mark, histone active mark, and then around 68,000 for repressive histone mark. And then if we did differential analysis, we had 600 ATAC up and 48 down. And around 1,500 the active histone mark up and then 1,200 down. We also had 795 down H3K27 and 1,200 up and down. So the RNA-seq we had around 1,400 genes upregulated and 1,000 downregulated. This is already published, and Thomas talked about it yesterday, where we compared Atlantic salmon and rainbow trout in vivo and in vitro antiviral responses. So we wanted to see if our data from different assays overlap. So we did see some overlap between H3K27AC and H3K27Me3. There were 21 regions that were overlapping between them, which is a small number. We expected that. We also had 10 between RNA and H3K27Me3. But we had majority of our peaks, we had around 200, were only shared between ATAC, H3K27AC, and had RNA-seq. We also had 171 regions that were in ATAC-seq and also showed H3K27AC, but were not present at gene level, so they were not transcribed. So I think it would be interesting to see our microRNA data to see if we find any answer for that. So I was interested in these 197 genes, which had open chromatin using ATAC, and then H3K27AC, and then RNA-seq. So we did some gene set ontology enrichment analysis. So these were the GO terms that were enriched. They were all to do with antiviral responses. We had an innate immune response, we had response to viruses, and we had some ubiquitination. And in general, it was indicating that we had those genes are involved in antiviral response and the activation of innate immune system. So next, I want to see the pathways that are involved in the antiviral responses. So induction of interferon-stimulated gene, which ISGs, which are the key antiviral genes. So I picked two pathways, RIG1 pathway, which is this pathway detects viral RNA, and then initiate this pathway where transcription factor IRF7 and IRF3 binds to the promoter regions of interferon type 1, and then interferon type 1 are induced, and then these interferon type 1 binds to the interferon receptors and initiate the JAK-STAT pathway, which in this pathway is initiated, and then this complex, STAT1, STAT2, and IRF9 is formed, and this complex binds to the binding site of ISRE, interferon response elements, and then ISGs is induced. This is a conserved region, and it's involved in the induction of ISGs. So we wanted to see if we can find the ISRE regions within the ISGs. So I picked here, just showing the STAT FAT3 genes that are involved in this complex, in the JAK-STAT, STAT1, STAT2, and IRF9. So we can see that I've only used one paralog for each. You can see that, so here, this is I picked the longest transcript, and the arrow is showing the direction of the sequence. And then this is the RNA-seq data, where we have the PBS and poly-IC stimulated. And on the top, we have the ATAC data, PBS, which is control, and poly-IC. We do see a clear differentially increased peak in poly-IC compared to the PBS. Then we have the H3K27IC. Again, we see differentially increased peaks in poly-IC, the stimulated fish. And while for the repressor mark, we don't see any peak at all for the poly-IC, but we do see a base level peak for the PBS. And then we found these ISRE sequences exactly lining on those peaks. For some of them, we had more than one regions of these motifs. So I call these functional motifs because they were on the open chromatin regions. But just to note that these sequences are everywhere in the genome. So I'm not sure the other ones that are at the promoter region, but they don't have a peak for ATAC or for the chip. I'm not sure if they could be called functional. So I've also done this for the RIG1 pathway, but I'm not going to show it here. So next, I wanted to look at these motifs, see if we can see them at see difference in paralog level. So this is MX, which is a key antiviral gene. It has 7 paralogs in salmon. And only one of them had ATAC and CHIP differential peaks. And while we have 2 which were differentially expressed at RNA level, but this one didn't have any ATAC or CHIP peak, and also, of course, didn't have any ISRE region enriched as well, but only this one, which had ATAC and CHIP peak for the CHIP active mark and ATAC, while at 2 ISRE regions. So I've done this again for lots of anti-viral genes. I'm just showing one here. Next, we also did some transcription factor binding site analysis using ATAC and CHIP data. And again, we have a ISRE motif enriched binding site for the IRF9, which is an ISRE sequence, was highly enriched in poly-IC stimulated fish. We also had BCL6, which is a B-cell differentiator. We had AP2s and the AP1 binding site family, and IRF6 as well. Yeah, we have lots of epigenetic data from for my ATAC and chip, but obviously we don't have time to present all of them, so I picked a selective one. To conclude, we were able to investigate antiviral response gene using using multi-omic approach. We were able to identify regulatory motifs involved in antiviral response, and we also found parallel-specific functional ISRE regions in key ISGs. I want to thank everyone involved in this project, especially AQUA-FAANG, for giving me the opportunity to do my PhD, and also my supervisor, Pierre's lab, and also Dan's lab for the help.
Thank you very much. Questions? We have plenty of time, so we can discuss a little bit about, yeah.
Thanks, Shamir. So with your ATAC and H3K27 acetylation data, it looks like you've filtered it basically to be a promoter based on location, right? Are you planning to look at any other more distal elements like enhancers with this? That was one of my questions. And then the other one was more on the immunology on the transcription factor binding sites. You have PRDM1 and BCL6, which I think are involved in B-cell development, which doesn't seem like an obvious thing after poly-IC very early challenge. So I thought that was quite an interesting finding. Would you comment on
We had some,
that?
also gene RNA level, we had some B-cell differentiator as well. So it could be the start of the adaptive response, a signal to start adaptive response. But it's quite interesting to see at innate level, 24 hours, these kind of transcription factor happening. Maybe involved in innate as well.
And are you planning to look at enhancers or candidate enhancers with the data?
Yeah, definitely in future, yeah. For right now, we're just sticking with this. But I'm finishing my PhD, so if you get time, I will definitely do it.
Awful excuse.
Any other questions? Okay, so thank you very much, Shamir. And our next speaker is Pierre Boudinot from INRAE. And it will talk about in vitro mutant models for functional characterization of genes of the type 1 interferon pathway in salmonids.
Good morning. So I'm going to speak about the in vitro mutant models, so the mutated cell lines that my colleague Bertrand Collet developed at the lab. Bertrand is not here today, but he was interested in making those cell lines to understand better the structure of the type 1 interferon pathway in salmonids. I'm not going to speak very long about the type 1 interferon pathway. We had already several speakers commenting this. I would like simply to mention now that as in all innate immune pathway, this is based on a sensing step, an initial sensing step by different families of receptors, inducing signaling and the expression, the induction of many effectors. In the case of the antiviral response in vertebrates, this sensing leads to the expression of a master cytokine, which is a type 1 interferon. There can be several genes very closely related, and then this would induce through the YAG-STAT pathway a number of ISGs. What is important to remember here is that those ISGs, so interferon-stimulated genes, they can be effectors, they can also be regulators, and they very often have a role in a positive feedback loop inducing again type 1 interference. So the structure of this pathway is rather complex. And it's important to remember that it's very redundant, which is nothing surprising because it has to be very robust in front of viruses that have many subversion strategies. So the loss of function experiments we made aimed actually at trying to understand better the role of key genes inside this pathway, and also more practically to try to generate cell lines in which viruses could be produced with a better efficiency, because obviously this is a limiting step for vaccine production. So how to do a loss of function experiment for this kind of genes? A long time ago, we tried with Jean-Pierre Levrault in zebrafish to do it with knockdown using morpholino, which is a very classical strategy, and we failed for almost all ISGs, probably because they are very strongly induced and the morpholino do not really succeed to stop the expression. Of course, the knockout in vivo provides a very good alternative, but it's very often rather long and expensive to do, even in zebrafish. And if the gene is essential, it can play many roles, it's lethal. So it does not always provide a successful model. So Bertrand actually wanted to set up a protocol to make knockout in cell lines, which may actually seem very simple, especially for people coming from the mammalian world, but in fish models, it's not so obvious. First, because the fish cell lines are not easy to transfect. It might be a very silly, practical problem, but it's a real one. And also because cloning is a bottleneck. Because, of course, when you have mutated your cell, you get a mix of mutated and non-mutated cells, and it's necessary to clone cells, and actually very few cell lines accept to grow when they are as one cell in a cell culture well. So Bertrand finally succeeded to set up a protocol. We have a Chinook salmon line that is permanently expressing GFP. We can transfect guide RNA and Cas9 to make the mutation of the gene of interest and of the GFP in the same time. And we get a bulk population. We can have a first check to check that the expected mutated site is really mutated in the chromatogram. And then for the cloning, we get, after limit dilution, some clones that are GFP positive, and some clones that are not GFP positive, like this one. And those clones are expected to have been mutated by the guide for GFP, and most probably also mutated for the other guide that should have been transfected in the same time. And it works rather well, so we have a good screening system to relatively quickly isolate the relevant clones that we can then validate after cloning for the presence of the mutation on the two haplotypes. I'm now going to go quickly through two examples of what has been done within this project. The first gene is the cytoplasmic helicase MDA5. So MDA5 is a sensor. It actually binds double-strand RNA, which is an important step in the replication of RNA, most RNA viruses. And this is one of the pathways leading to the expression of type 1 interferon and then ISG, then the antiviral states in the cell. So we used one guide. We could verify that in one of the cell lines we had a deletion of 2 nucleotides in the expected site, so it was really mutated, and in another control cell we had just a substitution that had no effect. And we used it for control. So the C1 is a mutated and the C2 is a control. In this case, when we infected cells with the alpha virus, the salmon alpha virus, we got almost 1 log, well, approximately 1 log, being optimistic, of replication, of additional replication, while the control cell line was behaving as a parental cell line, and which was completely consistent for the expression of one of the type 1 interferon, the A3. We had a normal, very, very high induction of this gene in the control line and a very reduced induction in the mutated line. Then we tried to check many other viruses, and I'm showing here what happens with the SHRV, that is a rhabdovirus, one of the these many rhabdoviruses infected fish cells. And we can see here that, again, the mutated cell line has an impaired induction of interferon Phe1 in IRF3, that is another key transcription factor of the type 1 interferon pathway. MX, that is a very classical effector, is also a little bit reduced, but not so much. This is one of the gene of the pathway that is not much affected by the mutation in MDA5. But what was also interesting to see was that we had actually no effect on the replication of this virus. It was rather similar between the mutated and the non-mutated cell line. And this is something we have actually seen in many of the mutated cell lines we developed in this project, which was not especially what we wanted, because we would like to have cell lines producing a lot of viruses, but which was not really unexpected because it's so redundant. So just to summarize what we have seen about MDA5, we got a mutated cell line. We have a quite clear effect on the induction of the genes of the interferon pathway. No drastic effect on the cytopathic effect that I have not shown, but it was true for many viruses. And a slight gain in this case for the replication with one RNA virus, but not for all the other viruses we tested. Second example is RSAD2, that is also known as viperin. That is an effector genes, typical ISGs, almost all the time induced after viral infection or interferon treatment. And the main mechanism known for this gene is actually the conversion of CTP into DDH-CTP, so it's an analog of a base that actually impairs the replication of viruses in many models, not all, but many cases. So we developed a mutant for this. We used two different guide RNAs, and we validated them in vitro before in vivo, and the reason was that this was in a cyprinid cell line. It was actually one of the tasks we had to do in this project to try to extend the protocol to other species, and so we did that in a well-known and well-used cyprinid cell line, the EPC line. So we wanted to know if the guides were working, and they were working. And then we used exactly the same protocol as well as the one I described before, and we got mutants. In that case, the viperin being very well conserved structurally, we could use a monoclonal antibody to show by Western blot that in the wild type, after induction with poly-IC, you have expression, but this expression disappears in the two cell lines, in the two mutant cell lines we could select. Okay, so we analyzed the transcriptome of these cell lines, and I'm showing here what we have seen for one of them. In this axis is the difference between the knockout and the control at the steady state, and here in this axis is the difference between the knockout and the control after stimulation with interferon. So we have the corresponding population of genes that are affected by the mutation in one or in the other context. And briefly, what I would like to mention here is that if we consider what happens after induction with interferon, with recombinant interferon, in the mutant and in the control, we see that it's almost there is almost no effect of the mutation. So the viperin appears really like an effector, and in this case, there is no obvious effect of the viperin on the induction of the other ISGs. Another thing that was interesting was that even if the EPC cell is not spatially close to an osteoclast, we could find at the steady state a clear effect on the mutation on genes involved in the ossification pathway. And that was funny because this gene was actually described in the 90s, initially in rats, as a gene involved in the activation of osteoclasts. So there is really clearly something affected at this level also in our mutant cells. And this illustrates also that the genes we mutate to learn about the interferon pathway can obviously have other functions that are more or less or maybe totally disconnected. And so just to conclude, we have generated a number of mutant cell lines in mainly with the CHSE, with the Chinook cell, Chinook salmon cell. So for the PKR, the helicase rigi, the two transcription factors that are very essential to the induction of type 1 interferon, RS7 and 3, MX, that is the myxovirus induced gene, that is an important effector, and DA5, and the one I mentioned in the cyprinid cell lines. We are currently working on the phenotypes. This is not very original. What is very important is that the effect of the mutation is very variable with different viruses. And the main lesson is that the system is so redundant that for the moment we have very few cases in which we have an effect on the viral replication efficiency. We almost all the time have a very clear effect on the induction of the pathway, but apparently there are other mechanisms keeping virus down, which is, of course, quite interesting. And the last slide is to thank all people who were involved in the project, Bertrand being really the main person in the development of these mutants.
Thank you very much. There are questions? Yes. Oh, okay. Thank you, Joe.
Hello. Yeah. Thanks very much for the talk. I'm wondering if you have any plans to combine any of the knockouts, so do double or triple knockouts to see if you can block those redundancies, or...
Yes, so yeah, obviously it's an important question. We have made tests to see how many guides we could combine. It works until 7 or 8. We should actually test more. We have project to try to validate guide RNAs and then to try to make some bulk mutation. And we also have in parallel succeeded to get an MX1 plus 2 plus 3 triple mutant, which will be interesting because then the whole MX should be knocked out. But yes, definitely yes. And especially to try to get cells producing more viruses, it might be an approach. We will see if it's enough.
So ready? Okay, thank you. So you have worked with a Chinook cell line. Have you considered some other cell lines to work with?
Yes, of course we considered. That was not easy because of the cloning step. And so the SHK typically doesn't like it. Ross, in Ross teams, one knockout could be produced, but it was really complicated, apparently, to succeed to clone it. Bertrand has developed a new trout line that is very promising because it was growing very slowly in the beginning, and suddenly one subclone started to grow very fast. So we have quite hope that this one will be possible to clone relatively easily. So then, yes, we would have then a rainbow trout. But Atlantic salmon, we don't have solution for the moment.
Any other questions? Yes, then.
I guess slightly related to Tim's question, how are you dealing with the paralogs or the analogs, the duplicated copies in salmonid? We saw from Shamir's talk, there are all these different MXs, for example.
So we try to choose our guides in the beginning of the transcript, not to have partial transcript that might be annoying. It's usually possible to find sequences that match 100% to the two paralogs or to the present paralogs. And that works really well. Usually when the guide is efficient for one, we got everything mutated in the cells we have checked. What is more complicated, in fact, is sometimes to find something that is specific for one paralog when we like to have different cell lines for the different genes.
Other questions? Yes, Luca?
Yeah, very interesting. I wonder, it's one by one, it's very long and painful step. Is it invisible in the future to do it like a more in a massive or parallel way? Because we have so many candidates, so many, well, species, but also many candidates, even within just one species.
Yeah, of course, there are people doing the screening strategies. That was not the choice of Bertrand in the beginning, but it's something that we could try to develop as well. Yes. Of course, a limitation is the number of on one side the number of guides that could work in the same time, and the capacity to subclone properly the cell lines we would get interested in. But as Dan mentioned, because we have several paralogs, if we like to understand what is the respective contribution, it's already complicated, one group per one group.
Okay, thank you very much. So we are perfectly on time to welcome our last speaker, Oscar Aramburu from the University of Santiago de Compostela. He will talk about a tarbot immune map.
I don't know if you can hear me right now.
It's slightly hidden behind there.
Oh, okay.
If you put it on here instead, that
Okay,
should be much better.
wonderful.
So that's it, yeah.
Here? Here better? Yeah.
It's okay.
So
maybe if you just go on this side, sorry. Otherwise it's hidden behind your clothes.
Okay, so here. Right.
Yeah, that should be fine.
Do you mean?
Okay. Well, let's go back on here. Tell you what, here is the best.
Thank you very much. So I have the very bad tendency of slowly lowering my voice as I speak, so let me know if at any point you stop hearing me, right? So as the final speaker of this session, I don't have many new concepts to introduce to you. Paulino yesterday gave you a broad introduction on what was being done in Tarbot in the context of the AQUA-FAANG project. Today I will be talking about our work in the immunomaps belonging to work package 3. So without further ado, This is the standard operating procedure that all of the partners carried out. Sorry. So they were developed by our partners at the Wageningen and the Aberdeen universities. So for the most part, we followed the same broad protocol, particularly for the in vivo stimulation, using poly-IC as the viral mimic for the viral stimulation, and finally a video extract that was kindly provided by our partners at INRA for the bacterial immunostimulations. In the case of the in vitro stimulation, we did need to make a few modifications for the standard procedure, namely to accommodate for the lack of enough leukocytes to conduct the experiment. The first one was we needed to pool by pairs the populations of the sampled fish, and then we needed to conduct a different chip-seq procedure that was the microinstrumentation assay. So for the chromatin assays and the RNA-seq, we followed the protocols that were provided by NMBO with slight modifications that were with some recommendations that were given by our partners at the University of Padua. So you already know the procedure, so no need to really explain both of them. We could include the 3 histone marks that were expected to be conducted in the chip assays, namely the 2 activation marks here, K for trimethylation and 27 acetylation, and the repressor mark 27 trimethylation. So after sequencing, we carried out the bulk of the pre-processing QC and peak calling using the NF-core pipelines. I'm showing here only the chip-seq one, but we did it also with the RNA-seq and NATAC-seq stuff. And here you have the broad numbers of what we included. This is a typo. In fact, it was 180, not 108 datasets. But yeah, you get the idea of the different datasets that we generated. So now jumping onto the results. This is the PCA for the transcriptomic analysis. As you can see, principal component 1 explains explains the difference between the in vitro here and the in vivo challenges. For the in vivo challenges, although they seem to cluster together, they form very distinct clusters between the control samples in pink, poly-IC in green, and Vibrio in blue. When we go to the in vitro, there is a big differentiation between Vibrio and the controls, but not that great difference when we are looking at the poly-IC in respect with the controls. In fact, most of the clustering here is done at an individual level. So each pool for the controls match with the same pool for the poly-IC data. So here in this table, you have the broad numbers of the differential expression analysis carried out with DSEQ2. And for each of these different groups, we conducted a gene ontology enrichment that you can see here. So in this bubble plot, we are representing the top enriched biological terms associated for each of the datasets. I guess you can see it here, but here this box represents the different in vitro datasets. This is the in vivo, and for this one you have the poly-IC down-regulated, poly-IC up-regulated, Vibrio down-regulated, and Vibrio up-regulated, and the same goes for the in vitro. So I think it's better to see the summary of what we are getting here in this box. So for the up-regulated, for the enrichment on the up-regulated genes, we are seeing 2 main trends. In the case of both Vibrio assays and to a lesser extent in poly-IC, we have seen a a lot of terms that are associated with RNA metabolism, ribosome biogenesis, and protein modification and transport. This is not to say that there is no enrichment in immune-related terms. It is that the immune-related terms that we're getting here are not as prominent, and they are not very specific. They are the usual responses to stress, response to stimuli, and so on. In contrast, with poly-C, we are not getting that many Go terms, but they are very much more specific. We are seeing the usual interferon gamma pathway, the TLR-mediated response, and the response to cytokine that we should be getting here. And in particular, for the Vibrio in vivo, we are seeing this enrichment in the term from syncytium formation. For downregulated, we didn't get much enriched terms for the in vivo. In the case of the in vitro, the terms were associated with metabolism, cell cycle, and cytoskeleton organization. So besides looking at these broad differential expression tables, we also wanted to look at specific overlaps between each different list. So what I want to focus here is the overlap between the upregulated, so meaning that both in the case of Vibrio and in the case of the poly-C stimulations, these genes are differentially expressed in the same direction, and also looking at differential expression in opposite directions in one immunostimulation in respect with the other. So in this case, I'm showing two Venn diagrams. This one is for the in vitro analysis, and this one is for the in vivo analysis. So looking at some of the boxes that I'm putting here, we can see some genes that caught our attention. It is worth mentioning that our group doesn't have a big background in immunology, so this is our interpretation from the genetic point of things. Namely, if we look, for example, at the in vitro condition and the common upregulated for the Vibrio and the poly-C, we get this pair of genes, so the two copies of SOX1, that we have found that are related to the downregulation of the immune response of the immune pathway to cytokines. So this, to us, suggests that at this point there is a tendency to the shutdown to the decay really of the immune response. And we have another few that have already appeared in, I believe it was in Costas and Serena presentations, like NOT2, STAT4, that is a transcription factor that is associated with immune response. Many members of the PRMT families, in the case of the in vivo common upregulated. So you get the idea that for each of the different comparisons, we are getting a plethora of interesting genes that we would like to go deeper on. So we will leave the RNA-seq stuff right here and jump into the ATAC-seq and ChIP-seq procedures. So here in this PCA, I'm representing all four, the ATAC-seq data and the ChIP data for the 3 marks. So these two diagonals separate perfectly the ATAC assays. This one is the case of the ATAC in vivo, ATAC in vitro, and here we have the ChIP-seq that we did for the in vivo stimulations, and the microchipmentation that we did for the in vitro stimulations. So this suggests that there is also a differential. The difference between the in vivo and the in vitro are exacerbated due to the different methodological approaches in the case of the ChIP-seq. And this diagonal separates the inhibitor mark, that is the 27 trimethylation, from the marks that are associated with the upregulation of genomic regions. So for each of these peak regions, we annotated them using Homer, and here you can see the broad related to if they land in exon regions, intergenic regions, intron regions, promoter regions, or transcription termination sites. And this, very briefly, we did the blacklist for the Tarbot chipsec data, incorporating the 21 input sequences that we could obtain for our experiments. Of course, the blacklist needs to be curated with more input data. 21 is very little, but this is just to demonstrate to you that, well, in this case, around 6-7% of the Tarbot genome is not, it's included in the blacklist. Most of it belongs to high signal regions for the chip analysis, and very little is regions that correspond to low mapability regions. So incorporating the attack chip and chipseq data, we did 2 chromatin state models. In this case, I'm showing you the model for the in vivo analysis, which fits well with the golden rule of 4 marks and 10 chromatin states. In the case of the in vitro, we tried to do the same with the 10 marks, but we found that the state number 9 and state number 10 were being quite similar to other predefined states, so we lowered the definition of these chromatin states and then went with an 8-state chromatin for this particular case. Just to mention, as we have seen in other presentations, that this is the way how we identified what should be the enhancer regions for each of these particular experiments. Basically, assuming that ChromaHM is not infallible, we also conducted the annotation of the enhancer state regions and only kept those that landed in intergenic regions and intergenic regions that consisted on just the introns. So those were the regions that we marked as putative enhancers for each of the conditions. So independently, we did the differential accessibility and differential binding assays using this program, DeepBind, that is almost similar to DSEC2. It's very useful in this kind of binary comparisons where you are just comparing the particular response from a pathogen to a particular control. So we did this for all the marks. The differential peaks were also annotated, and to identify the promoters, just looked at those that were annotated as promoters or TSS. So here we asked ourselves if there were genes with differential activation signals in the promoter regions that were also differentially expressed. This is not necessarily the case, especially when we are looking at the immune response. There are a lot of genes that are, even in the controls, there are reports of, in other works in human and in pig, where the regions of the promoters are not differentially open. This is what is usually defined as the bivalent or poised states that you have maybe already heard of. But we wanted to see if there was some kind of correlation between both of these. So here we have a very ugly, table that shows the results of a hypergeometric test. That basically what we are doing here is for a given population, and our population is the complete list of genes that belong to the transcriptome of head kidney, and all the genes that are showing openness or activation signals around the promoter regions. We are looking if in that given population, in 2 different samplings, which are, for example, the sampling for the differentially binded promoter regions and for the differential expression expressed genes, there is some kind of overlap between both of them. So what I'm showing here, the numbers that are outlined are the the particular cases where this hypergeometric test was significant. So it's worth mentioning that this kind of test shouldn't be done with very little samples in one or other of the conditions, like here. But as you can see, for the case of Vibrio in vitro and in vivo, for ATAC and H3K for trimethylation, the upregulated differential binding and differentially expressed genes are highly consistent. So now, with the definition of what we consider the putative enhancer regions and the promoters with that differential activation marks, we wanted to explore if there was an enrichment in specific transcription factors, and if that enrichment was if there was a prevalent enrichment in immune-related functions. So in this case, we used another functionality of Homer to call the enriched motifs. To be as consistent as possible, we searched for the transcription factors that were annotated in the turbot ensemble assembly. We kept those and conducted an ontology enrichment using this tool, Metascape. So basically what you are seeing here is just an example of the combination of the putative enhancer regions and promoter regions. We generated this kind of data sheets and did this clustering. And here you can see all the different functions that are associated with the transcription factors that were enriched and located in the Tarbot assembly. Some like, for example, the signaling by interleukins, myeloid cell development. There are a few that are correlated with immune functions. And this one here is a protein-to-protein interaction network that is done by ENCODE, and the two most prominent clusters that we get was this one that was related with the positive regulation of transcription, coinciding with what we were getting in the case of the differential expression analysis, and this cluster of immune-related functions, which is response to oxidative stress. Again, this is the example for in Vibrio and vivo. We did it for the other groups. And finally, just to show an in-depth view of particular regions, we asked, okay, so we have found motifs for particular transcription factors that are enriched. What's happening with the genes that code for these transcription factors? Are they differentially expressed in the same condition that we're observing them? Are they showing differential accessibility or differential histone binding? So this is a summary of this intersection. Basically, in the second column, I'm showing the transcription factors that have that differential binding in their promoter regions. The second is the same, but for differential expression. And the ones that I'm outlining with this black line are the ones that are shown in that are differentially binded, and differentially expressed. And finally, from here, I wanted to select one of them, this in particular, FOXO1, to just look at what is happening in the neighborhood of this particular transcription factor gene. FOXO1 is involved in mucosal immune response. It's reported to regulate the expression of antimicrobial peptides, so it's highly specific of the bacterial immune response, and it's a promoter of phagocytosis. So here I have the results of the differential expression analysis. As you can see, in the case of Vibrio, there is differential expression when we are comparing it with the controls. It's not the case with poly-IC. And here is the IgG visualization for each of the in vitro conditions. So here are the controls, the poly-IC and the Vibrio. We are seeing the attack peaks, H3K for trimethylation peaks, and the chromatin states, and same for each one of these. And as you can see, in the promoter region of FOXO1, which is right here, we have very defined peaks for ATAC and H3K for trimethylation that are not shown in neither of the other ones. Moreover, this is a little bit harder to see from from behind, but if we look at the chromatin states, we have a broadening of the strongly active promoter chromatin state around the Vibrio challenge that is mostly missing in the other conditions, and high presence of attack islands along the gene. Just finally, just to point you out that here we have an attack peak and an H3K27 acetylation peak that matches with an enhancer that is differentially binded in Vibrio in respect with the other ones. So just to finish now, these are the main conclusions and the final take-home message that I wanted to give to you. So here we present the first functional annotation map in Turbot that incorporates data of regulatory elements in both leukocytes and head kidney in response to viral and bacterial mimics. We have generated 2 separate chromatin state models and carried out the differential histone modification and accessible region analysis. And particularly for the open chromatin regions and the H3K4 trimethylation, we have seen that there are major chromatin changes that are happening between the stimulations and the controls, particularly in the case of Vibrio. And finally, just to show that the next steps that we wanted to go ahead with is the intersection of the data that we have been generated here in the immune maps with the data that is, as Paulino said, that is being generated for causal genetic variants that are, in this case, associated with disease resistance, and also with data that has been previously generated for major QTLs associated with Aeromonas salmonicida and resistance to Aeromonas salmonicida and the VHS virus. And finally, a big thank you to my group for the technical and emotional support, to Professor Jesus Lamas for helping us into doing the immune stimulations, and to the folks at Dan McQueen's lab that really helped us kickstart this analysis. And also thanks to all of you for your attention. And that's it.
Thank you very much, Oscar. Thank you. So, questions? Yes, Luca.
So, Oscar, thanks for the talk. When you say about the annotated transcription factor binding motifs in Turbot, has been done some experimental work to do that, or it's in silico?
No, it's completely in silico. Yeah. So basically, with Homer, we get the transcription, the binding motifs for transcription factors that are, you can narrow it for only vertebrates, which is the case that we did. And you get the gene symbols for these particular genes. So then what we did was go to the ensemble annotation and checked for correspondence with that. So in the case where we were getting both copies of a particular gene, we were including all of them, but it was completely in silico. Yep.
Yes.
Hi, thank you very much for the talk. It was just your PCA results, the clustering, I guess, with the in vivo. This looks quite unexpected compared to the number of differential genes that you found.
Sorry, so what you mean?
With the RNA-seq.
With the RNA.
And I guess this is what Paulino was relating to when he asked yesterday about mine.
Yep,
yeah, so all these on the right, these all look very much like one grouping, don't they, rather than... It's quite a lot of differential genes reflected there compared to the PCA.
So the thing, I should have put it here, we didn't put any filtering for the fold change. So here is a fold change of 0. So there is no the differential expressions that we are showing here are based only on the fold discovery rate filtering of 0.05.
Oh, right.
Yeah.
Okay.
So maybe if we went for a fold change of maybe 1 or -1, we could see lower. But we wanted to reflect all the changes that were significant in the case of expressions.
Any other questions? Yes.
Thanks. It was a really comprehensive talk. I think you explained the application of the data quite well with a few examples at the end. I've got maybe an annoying biological question.
Oh, let's see.
So I've noticed this in a couple of the outputs, so I don't think this is particular to turbot, but the number of DEGs following poly-IC administration in vivo seems much higher than in vitro.
Yes.
That's
yes.
not very intuitive to me. Do you know why that is the case?
Not really. In fact, with the poly-IC immunostimulation, we are a little bit conflicted because at the same time we are getting, well, it's not as high, but we are getting differential expressions, but we are not seeing such a prominent differences at the chromatin structure. So right now we have two hypotheses. The first one is that the poly-IC simulation didn't work that well. The only one which we have discussed with Jesus Lamas and Paulino, with Carolina Tafalla, was that maybe what we are looking at 24 hours post-infection is maybe the last remnants of that initial innate immune response. We are not immunologists, so this is just speculative from our side. So we would like to know what your opinions are on this. But yeah, I don't have a proper explanation for.
If I could ask a quick follow-up. So I was thinking the last hypothesis that you gave is exactly what I had in mind. Mm-hmm. I guess you could check that pretty quickly with some qPCRs over a time course. Do you know if this was done before the experiment or if that data exists somewhere?
I don't think it exists. I don't know if Paulino can. No, I don't think so.
We don't have samples of the whole process from the initial of the challenge and the You are telling about checking across the.
Well, just a couple of genes.
Just a couple of genes.
Yeah.
Across the process.
Just a time course to see if you have missed the peak or if it's. Yeah. Yeah. Okay. It's retrospective.
Mm-hmm.
That's all. Yeah. Okay. Thank you.
Yeah, thank you.
Thank you very much, Oscar.
Thank you.
Thank you. So it's lunchtime. I would like to thank all the speakers of this third session of the AQUA-FAANG. Yes, Joe has some...
Yeah, just a couple of announcements before lunch. So for anybody joining the online stream, the stream for today is now closed. We have a short informal discussion session after lunch, but that won't be streamed. So for everybody in the room, please come back. We're running a little bit late, and I think we want an hour to set up for that. So if we come back at 2:10, please. A couple of other things just on behalf of Charla, who has gone above and beyond and made an AQUA-FAANG game that she mentioned during the dinner last night, and you can access it via a QR code which is on a screen in the corner of the room where we're going to have lunch. Everybody should give it a go, not just because it's fun, but because there are 3 prizes in the form of delicacies from Norway, Italy, and Scotland, I think. Also, Charla has made a really nice results brochure, and there's a lot of them printed. Please feel free to grab lots, take them home with you, give them to anybody who might be interested. Help spread the word. So yes, see everyone back here at 2:10. Thanks.